All right, why don't we get started? So welcome everybody. Today's webinar is all about key challenges of data-centric QSP and ML AI, scope of problems, when and how. And today we're going to have 4 speakers: Alexander Lukanov from BIS Global, Michael Monin from Biogen, Maxim Kodymchenko from Verisim Life, and Professor Murali Ramanathan, sorry Murali, from the University of Buffalo. You know, this is an extremely important area in drug discovery and biopharmaceuticals. It is that point where a lot of things have to be done, either calculated mathematically or explored to get ready for either preclinical or clinical trials. And it is a very important area where many companies have to put things in order to make those next steps. So I think today's talks are going to be very interesting. So for rules of engagement, so we're going to address questions during or after the individual talks. Now, each speaker has about 40 minutes with 10 minutes of time for talks. We'll also do talks at the end in a roundtable. So based on some of the questions we got, we may explore things a little further, etc. I'll moderate that. You can ask a question at a specific time in the talk, and if I see it and we can ask the speaker to engage that. The only thing we just have to be careful of is that we don't go over, you know, the allotted time, because that'll make it difficult for others. So I'll do my best to manage all that. So you can put your question in chat or Q&A, and I'll keep an eye on that. In the Q&A part, we're going to moderate around. We'll ask the different speakers questions and have their turn to answer them. The panelists and the speakers can ask questions, and then this is being recorded, so it'll be on demand afterwards. If for some reason you have to leave early or you missed the beginning or a part of the webinar. So for some of you that are here that maybe aren't experts in this space, I just thought to put together a couple definitions to kind of orient everyone. So pharmacometrics is a discipline defined as the science that quantifies drug, disease, and trial information to aid in efficient drug development or regulatory decisions. Pharmacokinetics is the activity of the drugs in the body over a specific period of time, including the processes by which drugs are absorbed, distributed, and localized in tissues and excreted. And then pharmacodynamics is the study of a drug's molecular, biochemical, and physiological effects or actions. It comes from the Greek words pharmakon, meaning drug, and dynamikos, meaning power. And then quantitative systems pharmacology is a discipline within biomedical research that uses mathematical computer models to characterize biological systems, disease processes, and drug pharmacology. So 2015 Visioneers is an organization that was formed after 30 years of working in the industry. And the part that I just wanted to mention here before I introduce you to our first speaker is that we're very concerned with fair data, you know, making things better for the scientists from a scientific method perspective, et cetera. And I've done a lot of work in this space over the years. And one thing that I've noticed is beyond the algorithms, beyond all that work, I personally have worked with clients whose areas aren't as organized as they should be, you know, from like even a model's organization perspective. So one of the things that I bring to the table is we help clients get organized in this space. And believe it or not, some of the tools that others scientists use in drug discovery could be used in these spaces and used well to help organize that. And so those are some of the things that 2015 Visioneers do from either a clinical pharmacology perspective or a DMPK, a PKPD. We help clients organize their data and their model environments to get the most out of their efforts and energy. And it's really important because sometimes you're at the intersection of clinical and preclinical, and sometimes people on different teams play roles on both sides, and that's when it really starts to get convoluted. And so helping companies sort that all out and keep it super organized, in my opinion, has been a worthwhile effort. So with that little speech that I just gave, I want to introduce Dr. Michael Monine from Biogen. He's a principal scientist and associate director in the clinical pharmacology and pharmacometrics. He has over 10 years of hands-on experience in drug development, including translational research and science. And he helps advance candidates from early stages to pivotal stages. Dr. Monine is an active contributor and reviewer of key journals in the field of quantitative systems pharmacology. And today his talk will outline typical PK/PD data and translational modeling aspects. So Michael, it's all yours.
Okay, thank you very much, John, for the introduction. So I hope you can see my screen. Just a second, so let me put it in the presentation mode. Okay, can you see it? Okay.
Yep.
Good.
Just display settings at the top, and then just switch it to duplicate. There you go.
Okay, just a second. Sorry.
No problem.
So is this
no, just go up to display settings.
Okay, I see it here. Okay.
Yeah, you're still in
yeah, okay.
You have multiple monitors. Okay.
Okay, now you can see me. Yes,
it's on its way.
Okay, perfect. Okay, thank you, John, for the introduction. So I will give a talk about essentially physiologically based PK/PD approaches in neurology, and I will also introduce a particular case study on intrathecally administered therapies. This is going to be an example of QSP approach that our company, and we are using basically to address drug delivery to the site of action located in the central nervous system. So this is just a quick overview of my presentation. So I will cover a little bit challenges associated with drug delivery and access to their targets in central nervous system. I will also go over some published physiologically based pharmacokinetic models in order to predict brain exposures in human. And then I will dive into intrathecal drug administration to enhance access to CNS and introduce special case studies of ASO drug delivery. And my presentation will include only published materials. Okay. Okay, let me first introduce you a concept of blood-brain barrier and drug access to the target sites in the brain, for example. So many central nervous system drug candidates fail because they cannot cross blood-brain barrier. Blood-brain barrier effectively isolates the CNS from the blood. As you can see in the cartoon here, so tight junction proteins connect endothelial cells, basically lining the surface of the blood vessels, and essentially preventing drugs or other molecules from penetration directly from blood to interstitial space in the brain or other CNS organs. Also, specific enzymes and efflux pumps, like PGP transporters, may actively remove drugs from the brain, as illustrated here. At the same time, we know that pharmacology of the CNS therapies may be driven by drug concentration at the target site. For example, in some, so here's a table just showing a few examples of neurodegeneration diseases and potential target location. gene expression or a protein that is associated with the disease progression. In case of Parkinson, for example, it can be alpha-synuclein protein or also called TLRC2 protein, which is produced in the midbrain and subtension nigra. In the ALS case, it can be SOD1 gene, C9orf gene, ataxin-2 gene, and some other genes which are widely expressed in the brain, cortex, and spinal cord tissues. And in case of SMA, it's predominantly in the spinal cord. So it's difficult to assess drug concentrations at the target sites. Even though we are able to deliver some amount of a drug, we are still not able to sort of quantify this accurately in order to predict the effect. There are only limited approaches to measure brain exposures. For example, those are biopsy and microdialysis, but these techniques are invasive and performed only under critical conditions. Sampling from cerebral spinal fluid, or CSF, is typically used as a surrogate. However, CSF does not really represent concentration inside the brain. Another opportunity is to use an imaging technique like PET and CT scans, but this technique is still rather qualitative than quantitative. So there is a high need in the novel methods that can actually adequately predict drug concentrations at sites of action in the human CNS. So here on this cartoon, you can see just an illustration of where the CSF is generated. It's typically generated in the place called choroid plexus in the middle of the brain, and then it circulates around the cortical area and then goes down along the spinal cord, and it's enclosed in the narrow cavity called the subarachnoid space. So typically models used to predict drug concentrations in human or in animals are based on compartmental approaches, and such a compartmental approach views body as a series of virtual tissues or areas, and extrapolation between in terms of concentrations or PK between animals and human are usually performed by allometric scaling. And these models are typically very empirical and relatively simple. However, in some cases, they can be accurate for some drug modalities. In contrast, physiologically based pharmacokinetic models started to emerge relatively recently, and they view body in physiological terms and using a priori or independent or drug-independent knowledge of the physiology, anatomy, and biochemistry. So this type of models provide a physiological basis for explaining relationship between structural and physicochemical properties of drugs, and also drug concentration dynamics, and of course the pharmacology effect. And this setting enables better scaling from in vitro to in vivo and from animal to human. There are a number of commercially available packages exist on the market now, so-called GastroPlus or SimSip. However, the field of CNS drug delivery still remains uncovered, mostly. So here I just want to introduce one example of a recently developed PBPK model for antibody disposition in human brain. And you can see, so this model is fairly complex. It includes 16 tissue compartments connected anatomically via blood and lymph flows. So monoclonal antibodies can enter. So essentially, in every tissue, antibodies can enter through the vascular compartment and exit via venous blood flow. So exchange rates are defined as, for example, first-order chemical kinetic rates, and essentially it's a mass balance model. Although very complex and they relied on a large number of parameters. This is just illustration of the parameters used in this model. So again, in this particular case, which is cited here, they estimated parameters for different species, and they basically built a mass balance equations connecting different processes, and that enabled sort of simulations. So first of all, developing the model based on the animal data, and then simulations in human. And the outcome of this simulation is illustrated here. So when they are able to predict the plasma concentration and CSF concentration, for example, of a certain generic antibody product. And in some cases, we can also extract the concentration of interstitial fluid in the brain. However, still information about drug access to the brain tissues, like deep brain tissues, may still be missing. Okay, so let me now switch to a special focus of this presentation, which is intrathecally administered antisense oligonucleotide therapies. So first of all, I will introduce why intrathecal administration is ever considered. First of all, it's the way to bypass blood-brain barrier. In this route of administration, the drug is administered into the CSF, cerebral spinal fluid, in the spine, basically in the bottom portion of the spine, as illustrated here on the cartoon. It's very similar to epidural injection. However, the injection site is slightly different. So instead of epidural injection case, which is done through the narrow space there where all the nerve roots are located, IT administration or intrathecal administration is done towards the CSF flow, which is located in the narrow channel surrounding the spinal cord tissue. So, heartbeat and breathing rates and other mechanical stresses, they basically modulate the frequency and the magnitude of pressure oscillations in the CSF, and overall, this effect may help sort of propagate the drug movement upwards towards the brain. Now, what are antisense oligonucleotides? So these are short single-stranded synthetic nucleic acid chains with this typical molecular weight about 6 to 10 kilodaltons, and the idea is to bind RNA basically in order to inhibit or increase production of certain proteins which are associated with the disease. Here is an example of, so on the left you can see an example of the ISO so-called gapmer, which is designed to sort of reduce generation of certain targets, genetic targets, and the names of these ASO molecules are tofersen in case of ALS and so on. They basically prevent mRNA from translation and production of a certain protein associated with the disease. Another type of ASOs acting according to so-called splicing modulation, and in case of SMN, ASO used to treat SMA disease, so-called nusinersen ASO. It basically replenishes the missing or broken SMN1 gene. Basically, it's restoring it, and that the system is able to sort of produce more and more SMN gene, which is missing in the diseased patients. So where we usually get the data in order to develop intrathecal ASO. So the main source of data is actually monkeys, non-human primates, simply because NHPs are monkeys are closest sort of relatives to human, and they have a similar geometry, upright position, and so on. So in the studies that we are referring to here, animals are sampled from plasma and CSF during the course of study when they're still alive, and and at certain time points these animals are sacrificed in order to obtain concentrations of the injected drugs in tissues. So based on this data, we built a mechanistic ASO model or physiologically based PK model for ASOs. And the cartoon here shows the structure of this model. So basically, the CSF flow or CSF cavity is introduced through this sort of middle column here. The injection, intrathecal injection, is done into the lumbar space, and then we specify exchange of drug concentration between other CSF compartments in the spinal cord, so-called thoracic, cervical, and up to the brain, proximal brain, and the distal brain CSF. And the CSF as a fluid, it has a sort of interaction, or it may exchange the drug with the adjacent tissues. In case of spinal portion, the drug can leak to the spinal cord, and in case of brain CSF, it can actually penetrate through the membranes and go directly to the cortex, hippocampus, cerebellum, pons, and other regions. So how we inform this model development is basically, again, based on the monkey study. So the compartments which are marked by this lollipop sign in this schematics here, we have essentially the concentration data for these compartments from the monkey studies. And in this model, we specify several types of parameters. So first of all, it's transfer rates, which are empirical parameters which are estimated based on the data. And volumes, those are physiological volumes that are introduced, like essentially they're fixed. So the data that we get from monkeys is typically basically it's linear with those. And here, this is just an illustration of this type of data for different dose levels. We get concentrations from a number of animals and just see that it's almost linear. That sort of simplifies the model development. At least we're assuming that that dose is not a factor here. I mean, it's dose linear, dose proportional. So the monkey data is typically very, very sparse, as you can see on the illustration here. So we are getting quite substantial information from plasma because we can sample relatively easily after each administration. In CSF, we can get pre-dose concentrations still in live animals. However, in tissues, we are not able to get time course for every animal. So we essentially, in tissue, we have only one observation, only one time point per animal. That actually suggests that we are not, when we estimate the parameters of our model, we are not able to estimate inter-subject variability. That means that essentially the only approach we can use is essentially just a so-called naive pooled analysis that would estimate the parameters as a sort of average parameters, assuming that every animal behaves similarly to other animals. Here is just a quick introduction into the optimization tools that we are using. So just an illustration, the three tools on the left, they are widely used population-based pharmacometrics tools used for non-linear mixed effect analysis. They are very powerful. However, we found that for the problem that we are dealing with, which is actually quite a over-parameterized system, ADAPT5 was the best choice because it allows us to work with large models, large data sets. I mean, large multi-parametric models, but we are not interested in estimation of variabilities. The only random effect will be here. It's just the estimation of uncertainty of the data and the model. So therefore, we're using ADAPT5 as a simulation tool, essentially as an optimization tool. So here, it's just an illustration of two models. Essentially, it's the same model with different parameterizations. So model number one provides parameterization in such a way that every parameter has its unique value and estimated. And in that case, we see standard errors are quite high. So coefficient of variation of estimates is above 50%, almost in every case. In case if we do a sort of more parsimonious model with less parameters, essentially assuming that some groups of parameters are equal, then of course we get a better picture in terms of uncertainty. Uncertainty decreases, of course. However, in this case, our model has less degrees of freedom. So I'm not going to discuss which model is better right now, because this is essentially, it's not the focus of my presentation today. It's just an illustration of the approach. So here, I just wanted to illustrate the comparison of the estimates of the predicted concentrations at the and the monkey data. So first of all, if you look at the concentrations predicted and measured in lumbar, CSF, and in plasma, we see that essentially distribution phase in the CSF lasts over 2 or 3 days with a sharp initial drop right after the intrathecal injection. And plasma PK follows the CSF PK within a couple of hours delay. And then long elimination phase detected in the CSF, right, is basically determined by the tissue half-life, meaning that essentially meaning that the penetration to tissue determines a sort of the residual time of ASO drugs in the CNS tissues and overall. And you may also notice that that concentrations in the liver and in the kidneys, they actually drop faster, indicating that liver and kidneys are the major ASO elimination organs. This is actually quite important picture, which essentially shows us a total sort of just the overall distribution of the intrathecal ASOs, and here we see that only up to 4 or 5% of the dose of intrathecal ASOs reaches its CNS tissues, total CNS tissues, including spinal cord and cortex and the whole brain. So it's only up to 4 or 5%, which may seem is not a lot, but essentially if you compare this to the other routes of administration like IV or oral, it's much more abundant exposure in the CNS rather as compared to administration via other routes. In case of, as we saw previously, in case of antibodies administered intravenously, we are getting something like 0.1% of the dose in the brain. In that case, here we get something like 4 or 5% in the brain, which is at least almost 50 times more exposure. So this is just to illustrate the mechanism of distribution. So essentially, after we give an intrathecal dose in the CSF, so most of the drug, so most of the ASO amount basically leaks to plasma, to systemic circulation. However, 3 or 5% goes through the CSF brain barrier and basically ends up in the CNS. In the CNS organs. So after some time, like a few days, so we observe an interesting behavior, as I mentioned before. So when the decay of the concentration in the CSF essentially follows the concentration decay in other CNS tissues. So meaning that the CSF concentrations can be a sort of surrogate measure of the exposure, at least a relative measure of the exposure in the CNS tissues. Of course, it's not always understood the ratio. I mean, between the CSF concentration and the brain concentration, but at least in terms of half-life, or for how long the drug stays in the brain or other CNS areas, CSF can be used as a good sort of indicator. So once we get a good picture or good understanding of drug distribution, ISO distribution in monkey, apparently we want to translate this to human. So translational methods are currently being developed. Currently, the simplest way is to simply scale the physiological volumes that were fixed for monkeys originally, just scale them by certain factors to sort of get human volumes in organs, like in different organs, like in the brain, in the spinal cord, and other tissues included in the model. So essentially, volumes, they are included as a sort of dilution factors. Another assumption is that the rates of distribution or kinetic rates that we estimated based on the monkey data remain the same in human. This is a pretty, it's a pretty strong assumption. However, based on the sparse emerging data that we are gaining from clinical studies, we see that this assumption is reasonable. So now once we get a approximate picture of human PK at the tissue of interest, so for example in the cortex as shown here, we can use other information about the potential of pharmacodynamics or pharmacology. For example, from a transgenic mouse experiments or any other data that relates to the concentration of the drug and potential inhibition of protein generation, for example. And it can be either direct model or indirect model, depending on the data that we have. And then we do projections in terms of decrease of a target engagement, so-called target engagement in the area of interest, for example, in cortex, and that actually gives us an idea about what dose we should select for the first in human studies in order to obtain a certain target engagement in the different areas of interest. So the caveat of this approach is that we don't know, in front we don't know the relationship between CSF biomarkers or the decrease in human and the actual dose. So this is essentially this information typically comes later during the clinical trials. And now I came to the final slide of my presentation. So let me conclude that that PBPK models can be viewed as a subset of QSP approaches in general. And developing this type of models usually requires a large amount of information, data, and knowledge. And the problem is that that this information is not always available, especially for the CNS therapies. Yet, these models enable identification of key processes and rates that drive the whole body distribution and especially access to target sites within the CNS in the brain. Uncertainty of these types of models still can be high, and we can hope that that novel techniques like machine learning and AI-based algorithms combined with traditional QSP or PBPK models can enable deeper utilization of data from other sources and basically enhance understanding of the drug delivery.
Michael, you muted yourself.
Okay, it's good. Thank you. So, okay, now I'm happy to take any questions.
So if you just want to raise your hand, I can select on you and you can ask the question, or you could put the question in the Q&A or the chat box, whatever is more convenient for you. And those are all in the ribbon for Zoom, either at the bottom or probably the top of your screen.
Okay, let me find that.
See. So while we're waiting for questions, if any of the panelists or speakers want to ask, go ahead. I was just thinking that in one of our last webinars, the concept of mouse models and the translation to humans was very didn't work too well in the grand scheme of things. And I think you've kind of pointed that out here as well. Could you speak to that a little bit, maybe?
Yeah, so in general, so physiologically based models, they are by definition sort of generic. They should not be like animal specific, right? So because ideally this type of models should include flows of liquids like blood, animal's blood, CSF, whatever, every aspect of the body, and then the penetration rate. In some cases, for some modalities, these parameters are known. However, not for all modalities. For example, in case of intrathecal administration and antisense oligonucleotides, this information is not available. That's, for example, in our case, we call this model PBPK model. However, it's still rather more empirical. And if you take this model and apply it for other molecules, it may not work. It may work or may not work. So some modifications need to be done. Okay. In that case.
All right, Max, you have a question. If you unmute, you can ask your question.
Yeah, please, Maxim.
Yes, sure. Thank you, Michael. And I have a question. So the first question is, very interesting subject. So my question is, did you use any chemical parameters or physical chemical parameters of them for your PBPK model? Because we know like block PBPK very important, but I guess you just avoided blood-brain barrier because you simulated intrathecal administration.
Yes, exactly. This is a very good point. So as I indicated, so this model is rather empirical still, and we do not take into account any chemical or biochemical characteristics of these molecules. However, we build the structure, basically interaction between the compartments in certain way that it reflects the prior knowledge of the distribution. If I can go back to this. Yeah, for example, here, so directionality of these rates, it sort of speaks for itself, it's based on the experience. So for example, we define only one, essentially only one directional flow from the CSF to plasma or central compartment, because we know that there is no transfer from blood to back to the CSF. However, there is an exchange between the CSF and tissues. And also the connectivity, the certain structure and alignment, it's based on a sort of physiology. Although, yes, the caveat is that this model is not a generic model. So in order to account for other drugs, like maybe antibodies, we will need to do some re-estimation of parameters.
Okay, got it.
There are actually other efforts involving computational fluid dynamics technique to understand the actual physical flow of the CSF. And that's another sort of another component that we need to be taken into account later. Mm-hmm. Although actually it's a good point too. So based on our experience, we found out that that under current condition of the injection, the distribution along the CSF is rather homogeneous. It gets basically homogeneous quite rapidly after the injection. So it may actually tell us that such a compartment level representation of this is quite reasonable.
Well, that brings us to the next question here that may be related to what you just answered. In terms of transfer of drug from IT site to brain tissue, you have divided the CSF space into subcompartments. Do you think the fluid dynamics in CSF can have significant effect on the distribution?
Yeah, so it's a very good question. So again, so this model is valid for certain conditions. However, if you want to change the condition, let's say the injection site, for example, if you introduce instead of lumbar site, we will inject into the cervical or the sternomagna, then the distribution may be slightly different. So then in that case, we will need to do additional experiment and reparameterize the model. So for that purpose, we actually define a larger set of exchange parameters. So as I shown here, so this is, so basically essentially one of the reasons why we specify this model 1 is it's because it's a sort of represents a finer sort of discretization along the spinal CSF, rather than in the case of model 2, we have just 2 parameters, assuming a sort of homogeneous, faster equilibration. So if there is a reason to believe that gradients emerge along the spinal CSF, we definitely can split each compartment into a larger number of subcompartments and basically solve this as a one-dimensional PDE, for example. And we have done that too.
Okay. And then we have one last question from Sasha.
Sasha, okay.
Thank you, John. Thank you, Michael. Very nice presentation.
Thank you.
Very exciting to see some of the interesting results. My question is, when you build that complex model, have you done any feature analysis on the model, meaning that all the compartments are necessary, and what would be the ranking?
Good question. Yeah. Of course, we did. We tried many different structures and different parameterizations, and I tried to play with the number of like a sort of uniform compartments, combine. Let's say, for example, make single CSF compartment rather than split it into 5 CSF compartments, and yeah.
So what would be your strategy, what was the criteria to decide one versus another? Is it simple, the deviation, the smallest deviation from the measurement points, or there is based on?
So I think numerically, we look at the AIC, which is Akaike information criteria, or other numerical metrics such as likelihood. Also the standard error of estimates to understand how uncertain the estimate is. things like that. And we also look at the profiles directly and visually, at least, if we are able to sort of capture the main phases. For example, we know that there must be sort of a multiphasic degradation, for example, right? So if you're able to capture the behavior qualitatively, then we are in the right path.
Thank you.
But of course, it's a sort of, it can be endless process, selecting models. So, and we have to stop at some point.
All right, awesome. Great talk, Michael. I think we'll probably wrap it up on that last great question, and we'll go to our next speaker. Thank you very much, Michael.
Thank you. Okay.
Okay, so our next speaker is Professor Murali Ramanathan. He's at the University of Buffalo, and his work is on the physiological effects and pharmaceutical treatment of multiple sclerosis, MS. Professor Ramanathan has extensive experience in pharmaceutical applications of big data and modeling analysis using machine learning and artificial intelligence, and large high-dimensional data sets containing environmental factors, genetic and immunological biomarkers, quantitative neuroimaging metrics, and clinical measures in MS. His talk will outline the aspects of big data-enabled, biomarker-based, AI/ML-guided QSP models for pharmacometrics. Professor Ramanathan, the floor is yours. I'll just stop sharing.
Am I good?
You are good to go. Thank you.
Hello, everyone. I'd like to start out by thanking the organizers for this opportunity to present our work. And I know my title is long. It says Big Data Enabled Biomarker-Based AI/ML Guided Quantitative Systems Pharmacology Models for Pharmacometrics. And at first sight, it might seem like I went into a jelly bean jar full of buzzwords and picked out a handful. But hopefully you're in the audience and you'll be able to sort of track along with these buzzwords and the points of contact and their points of contact in the work that I am going to present. And as John pointed out, my work is really at the intersection between computational modeling and simulation methods, pharmacometrics, and pharmacometrics, both from what I would consider a drug discovery and development perspective, as well as the bedside clinical perspective, partly because of my interests in multiple sclerosis. Here's my disclosures. And so what I want to do is start out with an outline that includes a very brief skeleton of background. My focus is really on the role of AI and ML in pharmacometrics, quantitative systems pharmacology. And I want to sort of highlight what are the opportunities and challenges in this space, and what are the strategies at the 100,000-foot level that we face. And so what I will do quickly after this brief background is jump quickly into two case studies that use some of the strategies that I'm going to talk about that are relevant to pharmacometrics and QSP. And both of these case studies, what I'm going to do is start out initially by presenting the biological context in which we approached some of our AI, machine learning, and data challenges. And the two and then I'll dive into the methods and the results and hopefully end up with conclusions. And I'm going to talk about two case studies. The first case study that I'm going to talk about is called generalized pharmacometric modeling. And the context in which I'm going to talk about is basically how do we leverage AI for modeling diverse populations. And the second case study focuses on disease progression models for chronic diseases and to use AI to sort of explore disease biomarkers that change during aging. And we'll get into the details of that in just a second. So let's jump into some background. So as you know, I started out with big data in my title, and there are a variety of operating definitions for big data. And typically one focuses on the number of dimensions, more than 50, sample sizes greater than 1,000. And then, of course, there's the classic V's of big data: velocity, volume, variety, and I think there are many, many other V's that have been subsequently added. And you can see that the big data space really benefits from AI and ML. And finally, I sort of, since much of our audience is in the drug discovery, drug development, and pharmacometric spaces, I want to note that many of the applications that we look at in our domain are relatively small data compared to what a traditional big data source, let's say Google or Facebook or Twitter, would be dealing with. We are dealing with small data compared to the scales, but our problems are actually quite unique. And the other thing that I've done, and I don't know whether all of you can see, that when you have very small amounts of data, there's relatively little that you can do with a great deal of certainty. And then there's this region that we like to think about as the power law region, where the training data set increases and the amount of learning that we get improves, our understanding of the system improves. And finally, I think this is something that I found quite informative, is to think about what happens when the amount of training data increases asymptotically. You reach a place where you see diminishing marginal returns, and typically you'll still see some irreducible error in this region. So this is a nice way to think about data sizes and how they affect our ability to learn or garner information about the system, complicated system of interest. Hold on. So there's a lot of jargon in the big data space. I won't bore you with the details, but typically when I use the term data point, it's a data point in high dimensional space, and you can represent it as a vector, x1i, x2i, where I is the ith case and 1, 2, 3 are different dimensions. Think of it as different variables on the same subject, and potentially in a large data set, each of these attributes, each of these x's, x1, x2, xd, may be a completely different data type. One might be a binary variable, the other might be a continuous variable, and another might be ordinal. We talk about the term dimensions, and that refers to the number of attributes of each point or case. So D is the number of dimensions. And those of us who think quite simply in terms of Excel spreadsheets or statistical spreadsheets, it's the number of columns of data. And then, of course, there's the buzzword metadata, and metadata is data about the data, who, what, when. And many of us in the statistical space are also familiar with the idea of a data dictionary, which is a repository or a separate file about the information on the variables in a database. For example, whether you measured something in nanograms per mil, picograms per mil, the data dictionary is a place you look at. Oops. So I want to start out by talking about the pros and cons of big data. What are the advantages big data brings to the table? What are the disadvantages? As I alluded to before, one of the big advantages of using big data meaningfully, I emphasize the word meaningfully, is that learning becomes possible. That you can learn a lot more about the system if you're able to use the data more effectively. Big data can be more diverse. That means that if you're interested in special subpopulations, because the data set is large, if it has been properly collected, it can have information on the diverse subsets that you're interested in. Rare events that you don't observe in small data sets may show up better. And of course, from a statistical perspective, you get better estimates of variability. Variability, standard deviation, is hard to estimate properly from big data. And with that comes the idea of reduced sampling error. What are the disadvantages of big data? You have the curse of dimensionality because you have a large number of variables, you run into the curse of dimensionality. The space, the hyperspace increases dramatically, and this causes a whole range of big data problems, if you will. From a statistical perspective, because you're looking at so many variables, so many dimensions, you have run into multiple testing issues that have to be addressed in the process of interpretation and putting the information together. You run into computational complexity, and then of course you run into issues related to collinearity, heterogeneity, and things that can compromise power. They may be redundant variables, and so gathering information becomes a little bit more difficult. So hopefully this slide frames the pros and cons of big data for you. So I want to sort of look at the space of machine learning and AI, specifically from a framework of pharmaceutical sciences, by which I mean drug discovery and development and pharmacometrics. And you can sort of see at the 10,000-foot level, what are the most common methods that have been used in the literature. This is a little bit dated, it's 2021, and you can see that there's a lot of work on supervised machine learning problems. Many of the papers, the majority of papers and publications have focused on QSAR, quantitative structure activity relationships. And in a sense, QSAR is low-hanging fruit, because many of the problems with respect to chemical structure space and molecular property space lend themselves quite easily to the strengths of machine learning. The problems in pharmacometrics, modeling and simulation are a little bit more challenging, and we'll get to that in just a bit. But this is sort of the lay of the landscape as of a year or two ago. So what I want to do is present a guiding framework that we in my group have used to help our thinking about using AI and ML in pharmacometrics. And what I want to start out with is by pointing out what are the strengths and weaknesses and needs and challenges that we encounter in pharmacometrics today. One of our challenges is the fact that we have smaller sample size. Our sampling is sparse in the scheme of things. We traditionally use compartmental models and other QSP models that are highly tailored and fit for purpose. And we use estimation and statistical methods whose main strengths are parameter inference, so that you can draw conclusions about the larger population from the sample that you have. The methods are generally things like non-mem and all these other methods that that do nonlinear mixed effect modeling are computationally intense. And the other part of this is that these methods are well established and proven for drug development, for dose identification, finding out more about the data that you have in a typical drug development setting. So those are the strengths. And then what you have at the big data set, and we've alluded to many of these already in the previous slide, I won't dwell on them. Large sample size, big data after all, less sampling noise, diverse and representative if it's nicely done. And so what we think about when we think about ML and AI in my group is that AI and ML is potentially a bridge between big data and pharmacometrics. And we have to realize that the vast majority of big data that we deal with today are data sets that are being independently obtained in the population settings and in the very general population or clinical settings. So these are sort of independently acquired, separate from the drug development process itself. So in order to work with big data, AI and ML methods are data-driven and autonomous. You don't need a very, very specific model like you do in pharmacometrics. They focus greatly on prediction and generalization, and they have to be computationally efficient because they have to handle big data effectively. So this is the overarching picture that both my case studies are going to build on. So what I'm going to do is talk about generalized pharmacometric modeling. And this is a phrase that we coined, and it's based on a paper that we published about 2 years ago. And I'll build out the case in just a bit for generalized pharmacometric modeling. Here's the reference in case you want to dive into the details. So I'm going to start out by providing the biological context in which we began this work. And you might remember that what we are trying to do as a group is to try to find out sweet spots, opportunities, low-hanging fruit for machine learning and AI to deliver fruit and benefits in the pharmacometric space. And we decided that one potentially interesting area would be to look at the issue of underrepresented groups and diversity in drug trials. And as many of you know, the US population is increasingly diverse and will only become more diverse in the years to come. However, when we do drug trials or even phase 1, phase 2 trials or obtain PK data, our samples are much smaller, and these samples are not fully representative of the population at large, the population that the drug, or even the population of patients the drug is intended to treat. And these disparities have got the attention of the FDA, and there's now a guidance on how you can, as a drug development scientist or as a drug company, address underrepresentation in drug trials. And basically, there are ways to design trials and do things differently so that you improve the diversity of the sample of individuals who join your trials. What we're trying to do is, in the interim, build bridges between existing diverse data sets and pharmacometric data. So that is what we are going to try and do. And here's the overarching view of our approach that we call generalized pharmacometric modeling and how AI fits into this. And so what I want to do is to, everybody see my cursor? So this is the typical pharmacometric modeling space. You have a structural model, you have a population model, and PK data is typically fed into the structural model and the population model. And then you include subject-specific data to develop a covariate model, and the covariates help explain more of the variability in the PK that you see. So what we are proposing is that we build out or enhance the covariate model as a starting point for bridging into both external data and AI. So what we are going to do is feed external data and the covariate model into an AI framework and figure out better ways to get a more generalized, diverse prediction of the covariates, and you can feed that back into your structural model, your population model, your covariate model, and you would get pharmacometric outcomes and treatment outcome profiles that are more representative than the current framework. So this is a method to think about how AI can play into traditional pharmacometrics. So this is a nice scheme. How do you go about testing it? And so because part of my lab works on the clinical aspects of cholesterol and lipids in multiple sclerosis, I became interested in cholesterol dynamics in humans. And there's actually a surprising amount of data on cholesterol dynamics in humans. What we decided to do was use a relatively old study of cholesterol. It's a compartmental model of cholesterol, and you can see there are 3 compartments. There's a production rate of cholesterol, there's a clearance of cholesterol, there's a distribution into 2 peripheral compartments, and this is the reference. And these authors did a surprisingly thorough job of exploring covariate models with the technology that they had at that time. And they had built out covariate models for the production rate, the size of the compartments, the total size of the cholesterol pool, things like that. And so we decided to focus on the elements of this covariate model and try to develop methods. And so here's the reference. It's an old paper by Goodman et al., Journal of Lipid Research. And here are the clinical methodologies. There were 54 subjects. And this is a typical group of people that might be treated with statins today, for example. And I cannot tell whether 1980 was the pre-statin era or the post-statin era, but it might actually be difficult to get a sample like this for a trial today, given the number of individuals in the US who are treated with statins. So this is our starting point for cholesterol dynamics. So now we wanted to focus on a big data component. And so what we decided to do was pick the NHANES study. The NHANES study is a very large biannual cross-sectional health assessment of adults and children in the United States. It's the National Health and Nutritional Survey. I think the E stands for environmental, I'm not sure, but it's conducted by the CDC. And what they do is they do this survey every 2 years, and they spend a great deal of effort making the survey representative of the population at large. So this, to us, was a very interesting data set. And the goal is they don't cover all the diseases that are of interest, but they sort of touch on the biggies that are of public health interest. And here are some of the things that they do. Here are some of the methods they use. There's medical data, there's dental data, there's physiological data, and there's a whole bunch of laboratory tests. All of them very, very standardized from year to year, cycle to cycle. So this was a good starting point. And so we first asked, as a scientist and with the freedom that we have for our time in academia, we said, you know, let's take a look at whether big data makes sense at all. And so we asked the data, asked the question, is NHANES representative of diverse populations? And this is a very open-ended question. And so what we really wanted to answer was, so this is a population-based survey. Can we answer questions related to specific diseases in specific subpopulations using this data in any meaningful way? And what we did, I decided to do was look at insulin sensitivity in Mexican Americans. And this is a very intriguing question because insulin sensitivity in Mexican Americans has been explored in a variety of epidemiological disease case control studies, the San Antonio Heart Study, the IR insulin resistance atherosclerosis study in the context of diabetes. And the bottom line is Mexican Americans tend to have far greater, far lower insulin sensitivity and far greater insulin resistance than even other Hispanic populations, white, black, and other race populations. And sure enough, what we found was we looked at measures of insulin sensitivity. This is done through what is called a HOMA-3 model. And we found that in the Mexican American sample that we had in NHANES. So this made us believe in the utility of NHANES for studying metabolic diseases and metabolic questions in general. And so what we decided to do was go into Enhance and look for salient predictors of the variables in the covariate model that was developed by Goodman et al. For cholesterol in humans. The first step that we did was random forest regression. I'll answer questions related to this method, but basically this is a decision tree-based method that uses an ensemble of random uncorrelated forests to draw conclusions. And we picked the first 15 most dominant variables that showed up from random forest regression. And these are some of these, and we did quote unquote validate many of these measures, even the included interesting ones like cadmium and urine specific gravity using other sources in the literature. So these were the features, if you will, for the next step. And in the next step, what we wanted to do was really build a model for how all of these 15 variables interact with each other. So the goal is really to have a data-driven QSP-like model and explain the interaction between the different features using the enhanced data. And here's the method that we use. We use Bayesian networks, and Bayesian networks essentially look at a data set and they make directed acyclic graphs. And they have some really nice properties. For example, these graphs are topologically ordered. You can draw lots of, and they look a lot like models that many of us who are familiar with QSP work. And here are some definition, but the basic mathematical idea is that we are decomposing the joint distribution. In this particular case, this particular joint distribution has been decomposed using this dicyclic, directed acyclic graph. And so you can basically get a QSP model. And so what I'm going to do is jump into some results now that I've given you the broadest outlines of the methods that we use. We use random forest regression followed out by Bayesian networks. And so what we did was we looked at the correlation between the random forest predicted values of these covariates, the production rate, the central compartment, the one peripheral compartment, and the total cholesterol pool. These are the things that Goodman et al. Modeled. And we looked in the context of the data they had provided for their patients. We predicted it using random forest regression, and we predicted using the observations in the enhanced data set, and we found a really good correlation. You can see we do quite well. And then what I want to do is show you the results of what Bayesian networks can do. As I said, the goal is to model the interdependencies. And you can see here, what I've done is taken the features that are associated with this associated complex of M1, M2P, M1, M3P, M total PR, and how they tie into all the salient features. You can sort of see which variables connect to what variables and how this complex disease process might progress. So this is the framework for actually building out data-driven QSP-like models using biomarkers. Hopefully you can sort of see I'm touching on some of the buzzwords in my title as we move along. So we've now got an initial bridge between big data to pharmacometrics, and our goal is to use such approaches to extend covariate models to diverse populations and to explore this idea of building data-driven QSP models from big data a priori, and then going on and applying these QSP models from the general population to disease-specific settings. So the next case study that I'm going to talk about is what I call biomarker-based systems pharmacology. And here you can sort of see the paper that we've drawn on. And the term that I'm introducing is a biomarker-based systems pharmacology. You've seen elements of this already as we took biomarkers from the enhanced data set and built a systems pharmacology model. But here our goal is to make disease progression models for metabolic diseases. And we are going to use some of the same elements, but in a different way. And the problem, the biological framework that we are interested in is to look at aging. And all of us here really know that aging has distinctive effects on cellular and biomolecular processes, and much has been learned about the specific aspects of aging in the last nearly 10 years since this important Cell paper that found key, I think these are 9 key biomolecular and cellular processes that are affected. I haven't listed all 9. I've just pulled out a few that are relevant to my interest in metabolic biomarkers, nutrient sensing, by mitochondrial dysfunction, genomic instability, and so on. But the other thing as we bridge across the scales is to realize that these cellular and biomolecular processes have consequences on regulatory pathways, multiple regulatory pathways, and these changes to regulatory pathways cause changes in physiological, psychological, and behavioral processes that we associate with aging typically. And then, of course, overlaid on aging is the fact that many diseases show age dependence patterns in terms of onset. And of course, metabolic comorbidities especially emerge in aging populations quite frequently. And of course, aging eventually affects independence and quality of life. And the other thing that I wanted to address as I tiptoed into this complicated problem was to see if we could get a handle on something that has always intrigued me. And that is, at least in metabolic diseases, dyshomeostasis in one physiological process can cause orchestrated changes in others. We all know this intuitively, but we typically tend to treat one condition at a time. So for example, we know that glucose dyshomeostasis in diabetes can cause changes in triglycerides. Here's carbohydrate metabolism affecting lipid metabolism, affecting cholesterol metabolism, affecting renal function, body weight, and so on and so forth. And of course, the fact that chronic comorbidities can affect treatment outcomes for other diseases as well. So this is the biological framework in which we are talking. And so you can sort of see I like buzzwords, and we call this all natural. And the goal was really to sort of model natural history in the general population. And here's how natural history can map to disease progression. If you have a good study, longitudinal or a cross-sectional study of the general population over time, or as a function of age, what you will see is the natural history of metabolic biomarkers turn into disease progression of diseases like hypercholesterolemia or diabetes in a subset of the population if the population study is well done. And we are focused on biomarkers, and we are defining a subset of metabolic biomarkers that I call pathologically relevant biomarkers. And there are many pathologically relevant biomarkers. Glucose is one of them. You know, I think it's a biomarker of health, but we use it to diagnose diabetes. We use it to treat diabetes. We use it in treatment trials. So it's a pathologically relevant biomarker. And our goal is to build models that are data-driven and biomarker-based. And here's our big picture cartoon of how we might do it. So here's population-based big data. You can have also affect, you can enrich your population-based big data with data from clinical registries, which are also getting bigger and bigger with time. And our goal is to feed this data into a framework for learning the salient predictors. And this does not have to be random forest regression, can be other machine learning, deep learning techniques. You feed it into a method like Bayesian networks that can create a QSP model if you're interested in QSP model, or simply skip both of these steps through a deep learning method that takes care of predictor dependencies. So now you have an AI ML framework for something that does the functionality of a QSP model for aging in populations. And then I'll talk about the next step, which is stochastic modeling. You can sort of see that I use hybrid methods a lot, because our goal really is to use AI and ML to bridge other techniques that are more effective at doing the clinical questions that we are interested in. And here, the hybrid component, the traditional technique that we are going to use is time series modeling, and we are going to use what are called vector autoregression models. And the reason for using vector autoregression models, these are time series models, is that they have an element of stochasticity. And these models take the joint distribution of all the time series, whether it's glucose and lipids and cholesterol and body weight at the same time, and they give you a covariance matrix that can be modeled. You can model a covariance matrix very effectively that tells you the orchestration and the crosstalk between these biomarkers. So that's really what we are trying to do here, at least in spirit. So this is the enhanced data set that we used, more details here. And what we decided to do initially was use aging biomarkers from that were identified from a paper by Crimmins et al. 2008. Here's the reference. And they looked at three groups on metabolic biomarkers. We also had another data set that was available to us called the metabolic plus leptin, because this was the same metabolic biomarkers, but also leptin, because leptin emerged as an important metabolic factor later during the study, and it was collected in a subset of NHANES. So we analyzed it separately because the leptin data was actually, we had very small sample sizes for leptin. So you can see this data set is about 6,400 individuals. This is 33,000. And we also looked at cardiovascular biomarkers because this was a subset of biomarkers that were identified as important, was an important cluster in aging by Crimmins. So we applied our techniques, Bayesian modeling, Bayesian network modeling, not Bayesian modeling, but Bayesian networks. And you can see that the Bayesian networks not only generate very useful interconnection maps between the different biomarkers, but they also tell you how much confidence you have in these connections and about the directionality of these connections. So there should be a few here connectivities that can be considered bidirectional because they're relatively weak. For example, here you can see that the confidence in the directionality is weak, but our confidence in the presence of a connection is strong. So it's really a very effective way to sort of critically assess your connectivities in complex networks. So here's our data from the vector autoregression models, and I'll give you some context. So what we've done is we've done Bayesian networks. We've built a network with the key features in the network. And because of the network modeling, we know the interconnection between the models. And what we do in VAR is look at the whole panel of 15 biomarkers, the joint distribution, if you will, and the evolution of that joint distribution over time. And the nice thing about NHANES is has data all the way from 10 years to greater than 80 years. And here's a graph of some of the biomarkers. You might recognize glycohemoglobin here in this particular subset, which is a biomarker of diabetes, total cholesterol, lipid biomarker. And so we looked at two different VAR approaches. Broadly, what we decided to do in VARs are very widely used in economics. And there are two main strategies for using VARs: restricted VARs and unrestricted VARs. The unrestricted VARs, what you might do is, let's say you have 15 biomarkers in your Bayesian network. You let the covariance matrix connect all of them in any way it pleases. So you have a very general, unrestricted covariance matrix. And in the restricted VAR, what we did was we limited the interactions in the covariance matrix of the joint distribution only to the patterns that were predicted by the Bayesian network. And we applied it to the test training framework, classic AI ML framework, 75% training, 25% testing. And I think all the graphs that I'm showing you are test data. I haven't shown you all the training data here, but basically we found that just as we guessed, the Bayesian networks actually performed restricted Bayesian network covariance matrix models did a lot better than the unrestricted open models that had greater flexibility. And the other thing that we were now concerned is one of the things with stochastic models is the potential for overfitting. How do you assess these complex multidimensional models for overfitting? And we decided to do this in a somewhat creative way. So here is what I call the annual sampling. Remember, we have about somewhere between 50 to 100 subjects at each year of age. And so what we decided to do was first evaluate what we call annual sampling. That means we sample everybody every year and we do test and training. And you can sort of see that our models do really well in this context. And we also did what was called triennial sampling. What we did in triennial sampling, the goal was to assess overfitting of our VAR models. What we did was we did the training on data that was sampled every 3 years. And then what we did was so there's, you can sort of see that these are the training points. For every 2 training points, there are 2 blue missing points that were not included in the training. And the idea was that if models are grossly overfitting, what would happen is you'd see gross overshoots at the points that were not included in the training setting, which is, you have 2 points that will help you assess that better. And we found that we didn't do badly at all when we reduced the sampling to 3 years. And then, of course, we went overboard. We sampled every 5 and 10 years. And we found that our VAR1 fits were generally visually quite good for 3 and 5 years, which is reassuring. But the performance were reduced with the 10-year sparse sampling situation. And finally, as I end, I sort of want to leave everyone with what I think is a useful thought and a useful philosophy. And the first is, I love the name of this theorem. This is an actual rigorous theorem. It's called the meal-free lunch theorem. And it is beautiful in so many ways. The mathematics is very interesting and it's all great. But it also emphasizes the fact that there is no universally superior learning method that performs all other methods. So that there isn't a panacea that is going to solve all our problems. We have to select the best methods, and the onus for selecting the best AI ML methods on us, pharmacometricians and QSP folk, is on us. And yes, simple methods can potentially outperform sophisticated methods. And so every time you jump to a more complicated, difficult to use or difficult to interpret black box method, you should invest in evaluating the value proposition for doing that. And no free lunch, it sort of encapsulates that whole idea in many ways. And here's my summary and conclusion. I want to point out that integration of AI into pharmacometrics and QSP presents unique challenges. AI can be an excellent bridge between big data and pharmacometrics. I'm making the case that hybrid methods are a promising strategy. Traditional pharmacometrics, nonlinear mixed effect modeling is not going away anywhere soon. And they might well be a necessity given the nature of the problem. I've shown you how Bayesian networks can help QSP-like structural models to be built with data and built them objectively. And the other two points are derived from the case studies. One is the GPM, the generalized pharmacometric modeling. And I've shown you that restricted bar models can be used to describe the orchestration of biomarkers over aging. And then I'm going to ask questions, but maybe I'll acknowledge all the folks I've worked with over the years. Most of my AI ML work was done by Mason McComb, who's now at Amgen, and Kelly Fellows was another student who started out going in this direction. And there are some of my long-term collaborators in MS on this slide as well. I have time. I wonder whether I have time for questions.
Yep, we got time for two. Thanks, Dr. Ramanathan. That was great. The first one was, can PCA be applied before Bayesian network construction to reduce features?
Absolutely. Absolutely. I think I'm somewhat agnostic about the exact methods that you use to reduce features.
Okay.
And so we ended up using random forest regression because it's a method that's very easy to explain to others, or at least because it builds on decision trees, and there's a very graphical representation that you can use, and it works across both discrete, binary, as well as continuous data. We were particularly interested in the continuous data features. I know PCA does that as well. So it's a feature reduction method. And I think that there's something to be said for Bayesian networks because they'll help you distill the information and see the interdependency pattern. But again, we are developing methods now that replace the entire random forest Bayesian network middleware, if you will, with deep learning. Right. Now, that's going to come with some advantages and some costs, and that's the comparison that we are in the process of doing. So I'm actually agnostic as to exactly what goes in there. I'm more interested in how do we get to the point of using big data? How do we get to using AI and ML to further drug discovery, development, and pharmacometrics at large?
Yep. Okay. The next question is, great work and great talk, Dr. Ramanathan. Can you elaborate on how we can leverage such models to aid drug development in metabolic disease?
Absolutely. And I think that the goal now, I sort of alluded to one of the approaches that we can use. So right now I'm working primarily in the context of NHANES, which is a population-based data set. So now from a big data perspective, we really have good registries that have good clinical data. So for example, if you're working with spinal muscular atrophy, so rare disease, you might have a data set out there of a large number of SMA patients. And you want to sort of enrich your big data sets with information from a disease state, or from a clinical trial of actual patients on treatment. So we want to build that bridge between the two disparate heterogeneous data sets and use it for better feature identification. So for example, if a disease is evolving, let's say after 45, many metabolic diseases show up after 45. The goal might be to use your treated population or clinical trial data set to look at the progression in the general population at 45 and beyond. And then, of course, we can start looking at other aspects of treatment. But you have to remember that getting at large treatment data sets is currently a big challenge, and that's really the rate limiting factor. It's going to come. You know, these clinical data sets are going to become increasingly available. We also have a huge shortage of longitudinal data sets that are representative. And so we have to cross all of these data challenges and data availability issues as we move along. I'm right now in the process of composing strategies because I sort of see that many of these data issues will resolve themselves in the progress that we are going to see.
Yeah, great, awesome, thank you very much. All right, that was a great talk, and there's a lot in there. Our next speaker is Dr. Maxim Kodymchenko. He is the principal DMPK scientist at VeriSim Life. He has a strong experience in developing AI-driven biosimulation platforms that enable pharmaceutical industries with the technology to accelerate and improve outcomes in drug development to advance human health. He also leads research and scientific efforts to make BioISM the most accurate predictor of PK, PD, and DDI profiles of xenobiotics. He will make the presentation about the use of AI-driven platform for prediction of drug pharmacokinetics following transdermal administration. Max, you're up.
Yep, sure. John, I can't share my screen because I'm disabled from screen sharing.
That's my fault. I missed you when you came in, so there you go.
Alrighty, as it works. I hope you can see my screen.
We can see it. You're good to go.
Yeah, great. Thanks, John. So, right again, my name is Maxim, and I'm principal DMPK scientist in VeriSim Life. And our company implements artificial intelligence methods for accelerating drug development and drug discovery process through prediction of drug pharmacokinetics, pharmacodynamics, and with some approaches to patient certifications. But before joining the company, I'd been working in academia as a pharmacology researcher and professor, and I participated in various studies in drug development, like in vitro experiments, animal experiments, even clinical trials, and we also used classic methods. And working within Silica company, I just realized that AI methods are the very powerful tool for pharmacology, for any field of pharmacology. And here I'd like to briefly describe my own research experience when we use our AI-driven platform called BioISM that implement mechanistics models and AI algorithms. So, and just for the start, so here we can see the main stages of drug development process that we usually used to use working in my lab. And you can see in with a blue arrow. And you see they're pretty long enough, like months. So some of them are even years. And with the red arrow, you can see that some stages with the help of in silico methods may be accelerated, and that will result in lower costs and shorter time for the drug to get to the markets. And preclinical studies are definitely one of the most important part of the drug development process. And it takes substantial amount of time and financial resources. And when you use mechanistic computational tools for PK and PD, and actually they have been used, I guess, for the last 2 decades. And they show that they have some weak points. They can reduce time, they can reduce expenses for drug development procedures, but the accuracy and translatability sometimes, I would say, is a bit poor. Excuse me. And when you use AI-driven approach, it can help increase accuracy of the mechanistic models and can dramatically accelerate drug development stages, in particular preclinical studies and clinical trials. This slide show how in our company AI-driven technology may be used in all stages of the drug development processes integrated with mechanistic physiologically based pharmacokinetic models. This algorithm associated with a drug database may use any information for prediction of the drug behavior in human body. The results may be based on the experimental PK parameters, or they may be predicted from in vivo data or in vitro parameters, as well as just from structural and physicochemical properties of drug compounds. The main advantage of the AI approach is its capacity to impute incomplete data. Thus, it can just fill missing values like physicochemical properties or PK parameters that they later will be fed into PBPK model and provide more accurate and realistic results. AI model can be trained for any targets, and thus it can predict any parameters necessary for PK or PD, or just knowing the efficacy of known drugs or drug candidates or any others antibiotics. Yeah, and here's our study. So in our study, we have chosen one therapeutic area, which is painkillers or analgesic drugs. Here we can see the main therapeutic area that is based on drug spending, and you can see that pain management is actually in the second group of drugs with like billions annual spendings. And moreover, it should be taken into account that many diseases, in particular, like in oncology or metabolic disorders, they are often associated with pain syndrome, and they require administration of analgesic drugs. And among the route of administration over here, transdermal drug administration, for example, is often confused with topical, but both of the route of administration usually related to epicutaneous application. That means getting drugs on the skin and then acting locally or going into the systemic circulations, just similar to the oral drug intake. For our study, we're actually devoted to prediction of the behavior of local analgesics that could be applied on skin, acting as topically or acting in a systemic way via transdermal administrations. And just a bit of explanation about our study. So we know that pain always indicates some problems in the body, probably disease, trauma, or anything else. And it is related to irritation or stimulation of specific peripheral structures that we are called pain receptors. They convert physical or chemical influence or thermal influence into pain signal and transfer them into the brain. And that's how we feel it. And we have some drugs, because actually pain is actually not a pleasant feeling and sensation. So we have some groups of drugs that can mitigate pain syndrome, and we use it for many diseases. But actually only 2 of them, opioid analgesics and non-steroid anti-inflammatory drugs, they belong to a group of so-called conventional analgesics. That means they primary focus or their primary activity is killing pain or reducing pain syndrome. And why we chose topical route of administration. But it's actually dependent on the drug influence. Analgesic are usually used as systemic agents. So they get into the blood circulations and affect target structures anywhere in the body. I would say across the whole body, in any compartments. And usually such therapy is associated with high incidence of adverse reactions. In particular, when we are talking about opioid drugs, we know that they have some capacity to induce, for example, drug addictions. They have some severe toxic signs, including respiratory center suppressions and other adverse reactions. But in contrast, when we use drug topically, that means we have or we expect of drugs to provide a local activity because of the direct engagements with receptors or efferent transduction ways without any systemic exposure. And that, of course, means that such approach would provide faster, stronger influence with dramatically lower risk of adverse effects. And moreover, sometimes using topical drugs, for example, if we are focusing on the receptors that are located in specific tissue like skin or adipose tissues, that I would say target engagement of drugs being in systemic circulations would be very poor or quite low, resulting in lower efficacy. For sometimes using topical administration, we can increase drug engagement because of very focused or local high local concentration of drugs just around the targets that we want to affect. So actually high activity and low risk of side effects, that are main advantages. Okay, and for this, just a bit of pharmacology. For this purpose, we have very powerful drugs. They belong to the group of opioid analgesic that are used for the pain management. And now we have like a great number of opioid analgesics being approved by Food and Drug Administration for the use in patients with pain syndromes. Of course, they have some limitations. They have some related contraindications. But nevertheless, there are some conditions, some diseases when the use is necessary. Mostly, like the first analgesics, they are of natural origins, but now we have some synthetic compounds. And depending on the activity and target engagements, we have some drugs, we call them full agonists. So they engage with all types of opioid receptors, and usually they provide greater efficacy, but unfortunately, they also have a lot of side effects and adverse reactions, mostly related to the activity effects on the brain structure and finally on the brain functions, including cognitive functions, of course. So just to reduce the incidence of or likelihood of side effects, there are some drugs that have slightly different mechanism of action. They are partial agonists, so they do not activate all types of receptors. They can activate only specific types, and that reduce the likelihood of getting some side effects in patients. But on the other hand, that results in reduced activity. So usually using different types of drugs, we're always trying to provide better health for patients and at the same time to reduce the risk of side effects. And they're using both of the drugs, we definitely see that probably we've heard from the news that opioid crisis was across the country, because regarding of the use of these drugs, regarding of the type of activity, we see some drug addictions and all the following side effects. So that's why we are looking for the new opportunities, and one of the opportunities is using them locally. For example, using them for transdermal administration. That was our focus. And why transdermal administration is so promising and so interesting. So here is the structure of the skin, the detailed structure of skin. So actually, the whole skin is composed of two main layers. The first one is epidermis, and another one is dermis, and they're quite different. And the main function of the skin and the layers is a barrier. They need to protect skin and everything is under the skin from the environmental factors that could be aggressive. and the very important top layer of epidermis. It is called stratum corneum, and it is usually considered as a separate layer because it serves as the main barrier for water and for many chemicals and chemical compounds. And I would say that stratum corneum should be considered as a separate barrier also because this is actually a dead tissue. So as you see, it looks like a brick and mortar structure. So we have just a lines of bricks. They are pretty firm and in a very compact way. And this is just a dead tissue. So it's really difficult to penetrate it. So, but nevertheless, some compounds, not all of them, but some compounds may penetrate through all the skin layers, including stratum corneum, and then can be used for transdermal administrations. Nowadays, mostly all of them come not in a form like gel solutions, but mostly in a more sophisticated devices for transdermal administration. For example, like here, they call patches. They can provide slower and longer drug administration or drug absorption through skin, providing more efficient and safer therapy. Why we say that they can provide more efficient and safer therapy? Because as you see on the right, in the lower part of the slide, here are the drug concentration after IV administration or oral administration, and we see the horizontal line. This is a curve showing plasma concentration of drug after transdermal administration, and it looks like IV infusion, because we definitely see steady state concentration because of the regulation release of the drug from the transdermal patches. And that actually very promising results, because as you see, using IV, we just see very high peaks that do not provide steady state concentration, or we can see the steady state concentration is much lower than we expected. That is why we usually say that transdermal administration could be very promising, except the problem with transdermal absorption, not only because it is not typical for any drug, but sometimes it's difficult to measure it in a living organism. And moreover, recently, just getting back to our case study, recently it was found out that along with some pain receptors that are located in skin layers, there are some opioid receptors presented in stratum granulosum and stratum basale, that actually two different layers of epidermis. That's why sometimes the pain may be suppressed just via these peripherally located receptors. And just the same way, if we can use a drug that can stimulate peripheral opioid receptors located in epidermis, it may provide analgesic effects with very low risk of toxicity, because we want the drugs to be in the skin without any systemic exposure. And that would be very important for the treatment of any skin lesions that may be associated with severe skin syndromes. But here there are some very important questions for physicians, for pharmacologists, for the MPK scientists. What would be the drug concentration in skin after topical applications? And what are the fraction of drug that reaches systemic circulations, and what would be the drug concentration inside skin layers? And also there is another key question: what would happen if skin integrity, in particular the integrity of stratum corneum, is damaged? And this question that are very difficult sometimes to answer using some in vivo methods or clinical trials. So we know that after transdermal administration, drug can penetrate, not all drugs, but some compounds can penetrate stratum corneum. As I mentioned, this is the most firm, the firmest layer in the skin. So if drug applied on skin, that is supposed to go through skin layer, enrich systemic blood circulation. And the main barrier for drug transport, as I mentioned, is stratum corneum, and it is barely permeable, and active molecule may go through it using at least three main routes. It may be sweat ducts, it may be hair follicles, or it may be just direct diffusion through stratum corneum. Sweat ducts is usually a way for polar molecules, and so they have low penetration capacity through deeper skin layers. So I mean, if drugs can go through the through the sweat ducts, this kind of molecules would stuck in lipid layer just under the skin and never reach the circulations. And that's probably some problem with accumulations and further toxic signs. Hair follicles also doesn't significant input into drug absorption because they are pretty shallow. Therefore, mostly drug molecules penetrate superficial skin layers, simply diffusing through it. It may be just either via lipid channels around the cells, but this way is a pretty long because it looks like a maze for the drug to get through it, or going through the cell membranes. But that means the drug should be very highly lipophilic. And if drug is very highly lipophilic, again, it may stuck in adipose tissue in hypodermis, just under the skin. And that is why when we estimate transdermal drug absorption, it would be very important to understand the drug behavior inside the skin, how drug behaves and how it can reach systemic circulation, and would be the fraction of drug reaching systemic circulation, and what would be the fraction of drugs that remain in skin. And for our case study, that also important to know what are the skin drug skin concentrations engaging with the targets, in our case with opioid receptors. So what we have created for this purpose, we actually use the pretty known PBPK model, very similar to the one that Michael mentioned in the first presentations. So it just composed of some compartments, indicated the main organs in the body, arterial blood flow, venous blood flow, connected to the oral gastrointestinal compartment, simulating oral intestine, oral drug administration, and further absorption. So what we actually employ using this system, so we created a better way of understanding drug and physiology all together using machine learning and deep learning. And so here is the main key way how we can marry, I would say, two engines together. So we have knowledge engine and research engine. So when we use knowledge engines, this is what already exists. That means for our PBPK model, that's kind of fractional volume for compartments, the fractional blood flow rate for these compartments, as well as some PK parameters for drug compounds that already know. And research is a prediction, interaction prediction, because some of these parameters are not known and they should be predicted by ML engine. And the results would be looking like that. That should show what would be the drug PK profile. And so what we can do to also increase the accuracy, because as I mentioned before, the accuracy is the most important point for any prediction of drug pharmacokinetic and drug pharmacodynamics. So we use a comprehensive framework. And here is a simple illustration how we work with incomplete inputs from any of those features. I mean that we are using mechanistic model for modeling of drug pharmacokinetic profile, but usually, many drug parameters are missing. So sometimes, even when we go to some public databases and proprietary databases, we are unable to find some specific parameters like protein binding or maybe microsomal clearance and other things. So if you don't know them, the accuracy of PBPK model would be quite low. And AI could be deployed to input incomplete data. In other words, when we use multi-compartment models, it serves as a knowledge engine, what we know, operating parameters with known and verified values. And when we turn on machine learning, it works as a research engine, providing some insights into what the values of missing parameters should be. And then we do the iterative process, or we can even say iterative feedback process between these two engines that can lead us to more accurate parameters. And when the model makes prediction, we can also identify the underlying mechanisms that aren't captured adequately and actually send it to the users of the system. And here there what we've got. With BioISM, we had validated more than 1,500 compounds with experimental PK profiles. and we uploaded them into the database and then used it for testing and for training of their artificial intelligence agents. And the results demonstrate that after testing and validating, here is the high accuracy level for prediction of basic pharmacokinetic parameters that usually used for any drug development study, like maximum concentrations, area under curve, mean residence times, and volume of distributions. Here is the graph showing the accuracy in predicting main pharmacokinetic parameters. They're pretty decent, and we would say that they are usually derived from incomplete data. And I will show on my next slides what do I mean when I say incomplete data. But getting back to our study. So when we are talking about transdermal drug absorption, you want to know drug absorption through skin, and we can use two types of models. Here is the first one, and this is a single layer model, assuming skin is just a barrier, a single barrier. And this model is good for calculation of steady state flux, also for the permeability coefficients, for diffusion coefficients, and finally for bioavailability. Because actually drug absorption would correspond to the fixed first flow. So the drug absorption rate would depend on the concentration differences, on the thickness of skin layers, and it will also depend on the square on which drug was applied. AI technology can treat the skin barrier as a bulk, predicting the whole transdermal absorptions on the basis of specific chemical descriptors. So it does not need to provide accurate information for skin rotation, but it can show what is the systemic exposure for the drugs going through the skin. But if we need to know what is the skin retention for drugs that would be applied topically, then we can use mechanistic model with 3-layer scheme. And it is more relevant because each skin layer, like stratum corneum, viable epidermis, and dermis, are treated as a separate skin compartments. And all absorption parameters, diffusion, resistance, permeability, can be calculated separately, but using the same equations. And in this case, AI model can treat each skin layer separately, predicting drug permeability through each layer, including rotation in each particular layer, and drug concentration changes in time. Generally, this model is actually pretty much the same as the previous one in terms of implementing drug parameters and predicting drug behavior in skin structures. On the other hand, a 3-layer skin model also allows prediction of drug transdermal pharmacokinetic in healthy skin. So we see the 3 types of equations showing the drug absorption through skin, but also in the case of skin lesions, because usually when we have the skin lesions that may affect drug absorption, that probably related to the problem with stratum corneum integrity. It may be just disrupted or it just removed, and in that case, drug absorption would be much higher. And for transdermal modeling, we can just remove equation that is reflecting drug absorption through stratum corneum and proceed with a prediction of drug absorption rate as we did it before, and it would show what's going to happen with drugs in a case of application on the wounded or injured skin. And usually such type of conditions are associated with pain syndrome. So we decided to test both of these models, and we have 2 drugs, oxycodone and buprenorphine. All of them are opioid analgesics. All of them are agonists of opioid receptors. All of them were shown to be good for using transdermally, because probably I need to mention that there are just a few of drugs that can go through the skin by themselves. There are about maybe 2 dozens of drugs approved by FDA that doesn't require any specific formulations and permeability enhancers to go through skin. But these 2 drugs, they can do it without any permeability enhancers. And you see the specific drug parameters, and you can see there are some values that are marked with asterisks, meaning that these values were not available for the drug that we used on the study. And therefore, these parameters, we needed them to be predicted. for us to use them in our model and use them in 3-layer skin model required for calculation of drug behavior. We know that most important parameters for AI algorithms are lipophilicity, which is log P, and dissociation constant, which is pKa, and usually it is available at experimental, or it even can be calculated, at least for pKa, using specific software or even AI-driven algorithms. And they actually define the main drug properties, and including the capacity to go through the skin or stratum corneum, and also all other structural data that can be used as the descriptors to predict the behavior of those drugs. But other parameters, like for example, tissue partition coefficients or transdermal permeability, those are really difficult to get obtained using in public databases, also using some in vivo animals. But we actually use them as predicted by AI algorithms in our study. So prediction, so AI model was trained using our proprietary database that contains 1,000 of compounds, including all FDA-approved drugs, and we use it also for validation. So for validation, we use the publicly available results that you can see on the screen, and you can see the main settings for those experiments that are given to the right of the slides. This is just data published in the journal articles, and we just selected for 2 drugs 3 routes of administration for all of these drugs, and we use the models that we trained. How accurate would be the predictions of the 3 routes of administration for these 2 drugs? And this slide demonstrates the results of our predictions. So we can see that there are black circles that show in the experimental observables, and we see red dotted lines that shows our predictions. And we would say that accuracy of the prediction is pretty high. I would say more that even from my own experience, that red dotted lines looks a bit more realistic, but that's probably because of the variability that we always observe during any experimental study or any animal studies. So we can see that our test drugs are pretty good for predicted by our engine. So we decided to proceed, and we use this calculation for prediction of the specific parameters and specific coefficients showing drug absorption through each layer of the skin. So we can see both test drugs and the partition coefficients between stratum corneum water, dermis and stratum corneum, and viable epidermis and stratum corneum. That actually could be used for the calculation of drug concentrations and drug absorption rate and flux through skin layers. Just the same, there were some permeability coefficients that were calculated using the same things, and resistance coefficients. So actually those data would be just enough to use the equations that I showed before for the 3-layer model to calculate what would be the drug systemic exposure and what would be the drug retention and skin. And here are the results, what we see that. What is the drug disposition in patients with healthy and I mentioned with compromised skins? As we decided that the model for the compromised skins would be just the removal of stratum corneum, assuming that the integrity of stratum corneum is damaged. And we can see there are some differences and very interesting differences. So in healthy person, buprenorphine and oxycodone, they have almost a similar profile. Here are the different scales showing the concentrations, but we can imagine that probably oxycodone has an even higher plasma concentration after transdermal administrations, but they are like in the same range. But what we also see that in the case of the skin lesions, we see that buprenorphine have almost the same behavior. So it just goes through the skin layers with almost the same, a little bit higher absorption rate. Whereas oxycodone, in a case of removal of stratum corneum, go through the impaired skin with really fast rate. It is much greater the one shown by buprenorphine. And that finally results in dramatically high concentrations of this drug in plasma. And as I mentioned, when we talk about opioid receptors, so that means just applying oxycodone on a healthy skin, we have a low risk of side effects. But if there is any problem with stratum corneum, the risk of side effects after using oxycodone dramatically increases. And yeah, that's a good result to know. And the next slide shows the concentration of drug in different skin layers after using it in healthy patients, as well as after using in patients with skin lesions. And right again, we see the differences between oxycodone and buprenorphine. We see that skin concentration of oxycodone would be pretty much the same for hours in a case of healthy skin. That means that stratum corneum for oxycodone shows works or serves as the real barrier. Oxycodone is barely permeable through the stratum corneum, but actually provides pretty decent concentrations in this layers. And I would like to remind you that these are the layers where opioid receptors are located, and that means that oxycodone would be good enough to use in normal skin and to engage with this receptor. But in the case of some damage of stratum corneum, oxycodone actually almost not found in skin layers. It is almost completely reaches the systemic circulations and remain there. And contrast, buprenorphine works slightly different. So buprenorphine doesn't stopped by stratum corneum, and usually it goes through the skin layers regardless of the lesion with stratum corneum. The concentrations in viable epidermis and dermis is almost the same in normal skin or in skin with stratum corneum removed. So that can show that buprenorphine would be much more effective and much safer for patients with skin lesions that may be associated with the problem with stratum corneum. Here are the results of our study, and I just summarized in my last slide. So we can use our platform to demonstrate the potential benefits and also drawbacks of topical opioid agonists for management of some topically presenting severe pain syndromes. And this platform helped complete missing PK parameters that are required for accurate PK profile modeling for different routes of administrations, including transdermal or topical applications. And completed PK parameters can be used for predicting transdermal absorption rate and skin irritation of drugs with different physicochemical properties based on these properties and PK parameters. And AI/ML platform may serve as a decision-making assistant for the therapy selection for patients, for example, with skin lesions or any pain syndrome associated with any skin disorders. And this approach helps to understand drug concentrations in skin lesions, which is essential for the relevant dosing. But unfortunately, such measurements are impossible to perform in clinical settings because we understand that if we have a patient with skin lesions, we can just, for example, thermal burns. That would be just a lot of bioethical reason not to perform any clinical studies. But here, using the computational approach, we can predict pretty easily and with a high significance what's going to happen if we apply drugs topically in a case of skin disorders. And yeah, that's it. And thank you for attention. I'm ready to answer your questions.
As before, just either raise your hand or put them in the Q&A. Go ahead, Sasha.
Thank you, Maxim, for interesting presentation. Very interesting approach. Couple of questions. First of all, let's start from estimation of your PK parameters. So are you using the molecular structure of your drug to make an estimate, and how many, if so, like, do you have like extensive database of those different molecules and parameters, and then you can make the learning and make the prediction based on that database? How does it things organized?
Yeah, good question. So the first thing, yes, we have a database. We have a proprietary database, and this database consists I would say maybe 20,000 different compounds, but the most useful one, of course, all FDA-approved drugs or drugs approved by other national committees. And we trying to use any publicly available data because, you know, some data are proprietary and companies, then when they do some registering, they do not show them. So for, I would say for about 1,000 drugs, we have a complete list of all parameters, starting from physicochemical parameters that are easily available. For example, we can use PubChem database, so we can find lipophilicity, we can find pKa, we can find other parameters that are necessary. We can find also pharmacokinetic parameters like protein binding, clearances, different type of clearances. So everything is in our database, and we use it for training, like you mentioned. So training and testing, and this database, we always get the new data into it, and we repeat our training. But as I mentioned, some of those drugs are missing some parameters. So then we can use this train system to predict those missing parameters for the drugs that don't have them. And yes, we tried different modes of operation, and the final result that we could reach, that we can use only structural data. So the structural data would be enough to have some chemical properties, for example, TPCA. Then we can predict block P we can predict pKa, and then use it for prediction of other pharmacokinetic parameters. So actually, this is probably the final goal to reach, that we have just a SMILE structure. We run the engine, and we have the PK profile. We are on the way. We have, I guess, a lot of drugs that we can do it. We're also trying to refine our system to improve the accuracy, but we're on the way. And now we have some pretty decent prediction for just from structural properties.
So if you don't mind, a couple of more questions. When you use the SMILE diagram, obviously you convert it to digital world, probably using RDKit. Have you done any analysis of the properties which comes out of the SMILE diagram, which is the most important influence the decision of the particular PK parameters? Is there something you can say, like the majority of the cases, that what drives the mechanism and contributes to mostly towards the prediction value, right?
Yeah, that's right. But you know, even when we say we use several types of SMILEs, it PubChem SMILEs, PubChem isomeric SMILEs, PubChem canonical SMILEs, RDKit SMILEs. But usually using PubChem utilities, we derive chemical descriptor from them, and their number is really huge. So we can say just working with the structure, we have some 1,000 of chemical descriptors that can be used for the model training. That's why we use AI, because that's actually any model is impossible to digest all this type of data.
And my last question, so when you built your model of the skin and the different layers, obviously you make sure the discretization. I'm just wondering, like, given the structure you showed, obviously you're applying on the diffusion process, Fick's law. What is your main assumption when you come to the simulating the barrier? Do you take into account, you obviously take maybe just simple 1D case, but you could also have a vertical permeability and transmissibility of the drug just parallel to the barrier. And what is the impact of that on overall prediction if you take it, from 1D, let's say 2D model?
That's a good question. So, from our experience, we think that the simpler is the model, is the better. Because if we use, for example, this 3-layer model, we have like 3 times more variables parameters than for the single layer model. So for example, when we use single layer model like that one, we want to know what would be systemic exposure. So that doesn't matter for AI algorithm, what is actually happening inside the skin. We need to know what is the drug concentration on the surface of skin and what with the systemic exposure. And that's enough when we just need to know what are the main parameters for transdermal absorption. And usually when drugs are administered transdermally, we are always thinking about systemic exposure. But when we know where the drug can remain, when it can stuck, then we understand that the composition of different chemical layers is different. So that's why we can use these 3 types of layers just to understand where a drug can stuck, where could be high accumulation of drug, where could be lower accumulation of drugs. That's why we use this systemic model. But right again, the 3-layer model, I'm sorry. But right again, we are using just just typical equations for it, and the parameters and values for these equations are taken from the AI algorithms. Here's how it works.
Okay.
Okay, thank you.
Thank you. Michael, do you want to ask a question still quick? I know you had your hand up.
Yeah, thank you, Maxim, for a very nice presentation, very interesting subject. Actually, Sasha already asked all my questions, and you already almost all answered. So yeah, my only maybe a small question. So you considered a range of drugs, or is this a small molecule predominantly, or have you considered antibodies, something like that, larger molecules?
Well, talking about this project for transdermal, of course, antibodies can go through skin. So this is only about small molecules. And moreover, I mentioned that I counted, I guess, 20 drugs approved by FDA can go through skin. All others require permeability enhancers. For antibodies, we used to perform such kind of projects, but this is slightly different PBPK model. So actually, because there is no partitioning into the tissue, so the only thing that we are considered about is blood flow and lymph flow, and their concentrations, and then how it goes through all the compartments. So yes, so antibodies is there another field for PBPK modeling and using AI, but specifically to this project, no, these are only small molecules.
Okay. And essentially for this class of drugs, your PBPK model remains fixed. The structure is fixed. Basically, you are sort of optimizing the parameters, running your ML AI engine and using some relationship from the
Yes, absolutely correct.
Databases. Okay, thank you.
All right, thank you very much, Maxim. That was great. Got some good questions in there. Our next and final speaker will be Alexander Lukanov, Sasha, as we call him. And he is the Global Scientific Director of AI/ML, Uncertainty Analysis and Quantifications at BIS Global. Sasha has over 15 years of hands-on experience in solving multidisciplinary problems involving interesting synergy of physics, mathematics, numerical analysis, optimization, AI/ML, quantitative systems pharmacology, genomics analysis, and system biology. He focuses on digitalizing the lab experiments, utilizing synergy between laboratory machines and software systems, leveraging artificial intelligence and machine learning methods for advanced design. His presentation is about AI/ML for reverse engineering, application of polymorphology, and deconvolution of signaling pathways. Take it away, Sasha.
Thank you, John.
Let me make sure you have, yep, you're good.
Can you see my screen?
Yep.
Okay, good. Right. Okay, so thank you, John, for introduction, and I would like to thank all the presenters. So when I was thinking what to present as a last talk, obviously I didn't know what would be presented before, but I think, you know, it's a good opportunity to actually summarize and talk a little bit about AML, data centric QSP, and then combination, as Morley says, like to build that bridge. So in light of this, you know, my talk will be constructed in two parts. First of all, I'll give you a couple of examples of successful usage of AML in different applications. We'll talk a little bit about kind of in general where I believe, you know, we could get the benefits of deploying machine learning AI for QSP in general and in specific application. We'll talk about the forward model, controllability, observability, and reconstructability of the system. Obviously, I will mention a few things because our webinar is actually called data-centric QSP and machine learning, when, what, and how. So obviously, it would be important to highlight where I believe, you know, things can be done using the machine learning, how and what actually we can achieve by applying different machine learning techniques. Obviously, already a couple of presentations mentioned, you know, the application of the machine learning, but I'll try to kind of make a little bit of the summary, and I think all those previous presentation kind of nicely fits to the framework which I'm going to show. Obviously, I would like to mention a few things about the uncertainty analysis and quantification, because that's an essential part of any predictive modeling, and especially when it comes to the machine learning, because at the end of the day, it's a statistical method. different to conventional deterministic modeling approach when you provide the number and then maybe associated error bar. So here we always will be operating in terms of prediction, confidence interval, and the probability of confidence, how confident we are in terms of predicting certain values. And that's essential part when you actually formulated the answer and you provide a certain analysis based into account that the method itself was by the nature probabilistic one rather just completely deterministic. And last but not least, of course, I'll show you a couple of applications. I was trying to find some application which is different from the conventional PKPD type simulation. And actually that will be related to the scratch assay of the skin. That will kind of will be in light of what Maxim was presented previously. And I'll show you how the machine learning can be used to actually identify some of the system biology question and trying to understand the basic mechanism. And that would help, to actually build a final model to predict and simulate the impact of the healing of the scratch on the skin, for example, using the different drugs. So first of all, I'll start with a couple of projects. Obviously, I would like to mention one of the projects that related to pharmacogenomics. So you can do a lot of things when you have really big data here. We are talking about analysis of the genome, different readings, different expression of the genes. It could be some other part of the regulatory network. And it's really big data by definition. Morley actually mentioned in his slides. And analyzing this data, identifying the different aspect when it relates to different disease is essential part. And using the machine learning, as well as a combination of machine learning and conventional bioinformatics tools, you can achieve pretty significant results. So in that particular project, for example, things was analyzed based on NSP macroarray data for the set of patients with different doses of drug administered. And we were able to build the statistically significant conclusion based on the AML analysis, and basically correlate SNP and dose of the drug to different PG values and make testing of the certain hypothesis. One of the examples which I'm going to mention later on in great detail, which is related to quantity system pharmacology, will be related to actually analyzing the certain phenotype. In that case, would be just the rate of the healing of the wound on the skin, and then trying to show like how the machine learning can help to use the drug as a perturbation mechanism to reveal some of the signaling pathway, which is essential part also when you build the PKPD model, when you have a clear picture overall. But also it would be also important to demonstrate what is the impact of the uncertainty on certain process, and how can you quantify that and take into account in the model later on. And last but not least, of course, when you do analysis of the different drugs, that could be based on the small molecule, as we already discussed, it could be on antibodies, but it could be also on some other important protein, which is a part of the, for example, inflammatory signaling pathways. And for example, CFI deficiency is one of the important aspects which needs to be addressed. And machine learning is actually used to solve, identify the particular residue for the mutation, improve the activity, actually to predict the activity curve overall, which will be part of the final PKPD type of simulation when you know the activity of the drug, and you can build more accurate description. Especially we are talking about the potentially modified candidate, which in silico you predict certain mutations, certain locations, and then you can run the oral analysis to demonstrate what will be the impact of that particular variant with the corresponding activity curve on overall performance of the treatment process. So, okay, let me now start with the analysis and a little bit of summarizing what we've done in the previous presentation. And for the simplicity, I will focus on the structural analysis and linear analysis. Obviously, things can be easily generalized to the nonlinear situation, but I think at this point, it's just important to understand where things can be implemented and what are the benefits of the machine learning. So you probably already realized it in a couple of previous presentations, so that we are looking at some sort of solving dynamic system, PKPD type of simulation, and they can be described by the simple ODE system. Let's assume that for simplicity is a linear one. So far what we've seen were anyway linear ODE systems. It could be many compartments, could be single compartments, as Michael or Maxim described in his in their presentations. And of course, the state vector X is just basically the one we are looking at. It could be concentration, it could be something else, depending what you're trying to build. And that particular equation obviously could be additional parameters which is related to control parameters. And there are some obviously studies where just, trying to understand, what should be the process to, let's say, inject certain drugs at a particular time, or maybe set of times, or maybe continuous injection based on the concentration of the particular things in the system. And that allows you to introduce the state vector U, which is, basically your control vector. Of course, we have initial condition of the administrated time of our drug. And what we can say at the moment, so obviously our operator A is the main operator, which could be mechanistically based operator, it could be physiologically based constructed operator, it could be even machine learning based constructed operator. So, and I will roughly describe, the connectivity between general formulation, what, for example, Murli talked in his presentation. But the bottom line, that operator itself can be split into 3 components. One is a conventional PK operator, and we've seen that in Michael's presentation. Could be also contribution from the PD, and then it could be contribution from the PG, depending how complex your system and which effects you want to incorporate. Again, this is for simplicity. We're considering the linear system. Things could be generalized in a nonlinear case, but let's just keep it simple. And last but not least, of course, we all trying to measure what is happening with our system when we design and monitor the performance or the efficiency of our drug or treatment process. And for that reason, we have a measurement Y, and we have a connectivity. Sometimes it could be very complex connectivity with our state vector and our control vectors. And of course, if you measurement a lot Y, and it has a big dimension, and let's say matrix C is a square matrix, you can directly invert that matrix and find what's the state of your system based on your measurement. In that case, you even don't need to solve ODE or solve any complex, forward model. You just simply invert the data and obtain the state. And that's kind of what Murli was showing in his approach, when there are some observations and you can make the conclusion about certain aspects by just basically operating with some measurements and some of the data without even considering, dynamic process or dynamic ODE in his presentation. So that's perfectly fine. And depending on the case, obviously, sometimes it's possible, but sometimes it's not possible. In most of the situation, our measurement is actually, in most of the conventional PK/PD type of problems, is very limited. It's basically sometimes merge average concentration around the tissue, let's say concentration liver and some other aspects, and you don't have even distribution, or even don't have enough time points when you produce the measurement. And that's what makes the complexity of all the process. Furthermore, we could have all sorts of measurements. So let's imagine that, we administrated the drug to one of our study species, let's say mouse. That's very common study lab species, and we do measure not only, let's say, some of the data, let's come, from blood or plasma or like some other specific area, but we also do the imaging of, contrast imaging of propagation of some things, and we could measure that in time. And by doing these things, we're accumulating the large set of data, which ideally you would like somehow incorporate into your analysis and make sure that your model or whatever you're building is for the model does not contradict all other sorts of information you have. And then the question, like, how can we do these things? Can we actually incorporate all this data and make sure that they're consistent? And that raises 2 things about what we call controllability of your system and observability of your system, which is very tightly connected to each other. So let me tell about a little bit for controllability, especially for the people who doesn't have like a background in the control theory. What does it mean? Effectively, what it means is about definition. So if you have a state x0 at a certain moment, let's say time equal to 0 at your initial time, and we say that the system is controllable if for the given finite period of time, you could have a design such a control parameters u, and that basically will lead to xt is equal to 0. So effectively, you can always maintain your position at 0. It could be a generalized, obviously, in more general cases, but the bottom line is that what it means that if you have desirable behavior of your system, the controllability means that you can always keep around the desirable behavior. So if the system deviates from the desirable behavior, you can always find the control parameters to push it back in the trajectory you would like to have as your solution of the system. And sometimes it's not always possible for some of the system, but sometimes it's possible. And what we want from the basically designing the treatment plan, so that, we want to have a very desirable effect after all. We want to have a controllability. We want to make sure that whatever we inject or deliver administrated to the patient achieves certain level of confidence and desirable trajectory path, especially for the treatment of certain disease. So, and mathematically, obviously, we know that the controllability related to the parameter B, matrix B, as I was shown in the previous slide, and A is designed using various approach. So if we wanted to check where we can control that system, we just basically need to check that the following determinant of those matrices multiplication is not 0. It's kind of very complex from a mathematical point of view, especially when B we don't know, and assume that the B is actually obtained by a machine learning network, because our control parameters could be very complex. In that sense, obviously it's very difficult to check everything, but with current state of the advanced control theory and mathematics, thanks for that, it's able to compute those things very quickly and make analysis where your system is controllable. And that can be used as one of the filtering factor where you build your compartments model correctly and where it is that model is observable given your set of measurement or control parameters. So when it comes to the observability, we can also formulate similar condition, which basically specify that the given set of output measurements, you would like to make sure that you can obtain the information about the trajectory path. And as I mentioned, if you measure exactly the same number of points as your state vector and your C matrix, and that equation is a rectangle, you can basically invert that matrix as it's invertible, and you can already give the answer, right, just based on the measurements. But in reality, obviously, we don't have the luxury to have such a measurement, because measurement is always more complex than the overall dynamic system description forward model. So therefore, we have a situation when we can check that rank, but again, our link or operator could be described by a very complex neural network, which connects the measurement and the state vector. And again, we can check these things and make sure that whatever we put in our PKPT forward model is consistent with the observability criteria. So there are some other less important but still important aspects, which I'm just going to briefly mention, which is related to constructibility, reconstructibility, and predictability. All those aspects are very important. They will affect, the selection of your operator A in your equation and giving the measurement you provided. And that's an important step to perform, which kind of allows you to say that, subject to the measurements you have in the data about your system, about your biological system or the process you are studying, you can guarantee that you're observable, controllable, and there are some other nice features you can guarantee. And that obviously can be used when you're trying to filter out which compartment models you need to put in place and study, given your measurement. Obviously, life is not that simple. I mean, obviously, there are many complex biological processes in the system. It could be and obviously, sometimes we can build a system which is controllable for part of the variable states, and for some other part, it could be uncontrollable. And that's very commonly appears, especially that we don't have a full knowledge about all the regulatory network within the system. That maybe will be addressed at some point, but at this moment, we know some part of kind of environmental space where we know like certain signaling pathways complete and fully described, but overall we don't have the entire information. So therefore, like we can definitely split in terms of the controllable, non-controllable, and most of the time we need to operate with that situation. It's still important to understand what is controllable, because at least that will be as an important step towards the analysis of the final results. In addition, it could be cascade of model, and in that case, obviously, in the cascade of model, some of the models can be observable and controllable, but in our case, it could be completely non-observable and not controllable. And that also is a possibility, and that has to be taken into account. So where's the machine learning can come into the game? Obviously, we already discussed a few, several applications, important applications. First of all, operator A itself can be constructed, and especially we talked about the missing key parameters, which can be derived from the machine learning as per maximum operation. when we do the measurements, and we can use the measurements to already figure out about the state vector, so which more or less was described in the earlier presentation. And of course, when Mikhail was talking about his model, he was talking about like what general description or type of approach to the PKD simulation, and what are the necessary steps and data need to be put in place in order to select one model versus another model, different criteria. And one of the additional effect where we can apply the machine learning is actually integrating those equations. So, obviously there are already existing tools and methods, some commercial license, open source, academic codes, etc., maybe in-house developed code, but there are also opportunity to actually use the neural network to integrate the system, especially when it comes to very large set of ODEs altogether. We are talking about, millions of ODE, for example. And in that case, machine learning will help, to quickly integrate this one using, pre-trained algorithm. I would interesting to discuss a little bit more in details the aspect where we concentrating at BISC. So we assume that, the forward model, PKPD model is given to us, and what we are trying to incorporate is more detailed analysis of the data around that particular study. So imagine that we have a set of the images, it could be EEG signal, and we're trying to incorporate this information into the PKPD modeling. So how do we perform that analysis? So we extract the certain features from those input data, and then we train the model, and we're trying to predict the state vector. So imagine that your state vector is a concentration in certain compartments. Let's say it's a 2-compartment model. You measure concentration average in the liver or in the blood, and then basically we're trying to map all our information to this one. That's what I called the forward machine learning model, which trying to shrink all big data which related to images and some other time series, or it could be like a signal, into something which directly participate in the PKPD model. The bottom line, we want to check where the system is controllable and observable. For that reason, we actually need to now invert that neural network we built, and then check the criteria I mentioned in the previous slide. And obviously, it's a tricky exercise. It was a lot of high confidence in what you're doing, but the bottom line, what I want to make sure that you understand that it's possible to do. There are certain algorithms you can construct the inversion. This one with some approximation. Obviously, it's not directly inverse matrix per se. If you're talking about linear algebra, because those are system more complex and not invertible in the classical sense, but in the sense that, you can verify the contributory observability, it's still possible to do. And it allows us to check where whatever given to us PKPD model is actually consistent. It's consistent with those measurements which are provided by some other source or information available or associated with the study. And if it is the case, we can use the forward model, and we can, perform the conventional analysis, build the solution, check the half-life, check, the dosage, minimal dosage. And that would be, some sort of the entire framework, which allows us to firstly check that it's consistent in terms of observability and controllability of existing data, and then B, perform the analysis. So now, in terms of the uncertainty optimization, it's important to realize that when you do the measurement of some of the variables, state variables, you measure them with error, and that error will define where some of the compartments need to be present or not. Because if you know that your error of the measurement, 20%, then you can argue that some of the compartment does not contribute enough to capture that sensitivity, or basically will be some sort of diminished by the other contributing in your PKPD model. And therefore, there is even no point to refine your model adding by new compartments. So therefore, effect of the influence of each of the PK parameters on the error itself, and the comparison, the measurement error with the error which comes from the uncertainty in your parameters is an important aspect to decide which term in your equation needs to be stayed, because it will be, meaningful and measurable, versus some of the term which, may be important, but because you are not able to measure accurately enough, those are terms will not play significant role. So if you want to keep them significant, maybe you need to measure with the accuracy of 1% instead of 20%. And that's important to realize. So for that reason, we perform usually global sensitivity analysis using the Sobolev decomposition and some other aspects that we've done that analysis to demonstrate which terms in the conventional PK/PD model needs to be kept and used for the analysis. So usually the main elements here is obviously Sobolev decomposition of the variance. You computed that using the global sensitivity analysis. You build the hypercube graph for all your parameter realization. You compute certain standard deviation, giving certain information, and basically based on the total sensitivity and then split of the total sensitivity in terms of S1, S2, S3, you can make a decision which one of the parameters should be kept and what would be the impact of basically relationship between your sensitivity of measuring the data and your basically solution and the variance of the solution given the uncertainty in your parameters. I'm going to probably skip one and for the sake of time, dive in straight away to one of the examples. It's related to analysis of the signaling pathways. And for that reason, we will use the perturbation mechanism. So the drug is actually used to perturbate the system, measure the response to different drugs. And then once we know how the drug is connected to, let's say, a human kinome or kinesis, we can eliminate drug via machine learning, and we can connect phenotype with the kinesis. And by doing these things, we can see which are the processes the most important in particular for that particular phenotype, and we can target it. For example, in that case, we are talking about the scratch assay of the skin. We can target those kinases with specific drugs or combination of drugs. And I'm going to mention how we also use the machine learning to build up the cocktail of drugs, which has a specific target, certain kinases, and a limiting number of targets. So let me dive in. So into the details. So we have a drug, we have a target. We obviously have a data in it's the data which shows the how do your activity of the kinases is changed as a certain concentration of the drug. So you can obviously formalize this in terms of the activity matrix, and then this kind of the information can be used to answer two things. First of all, you would like to understand which are the kinases are most important, for example, for healing the wound, and also it is important to answer the question where it is possible to build a combination of two drugs at certain concentration, which will be more effective than one drug on its own. And that will be obviously related to polypharmacology domain, where people combine different drugs with different concentration to address more effectively one of the particular signaling pathways and treat the disease. So obviously for that reason, we need to make sure that activity curve is fit nicely, because we extract the certain activity out of this curve and then goes into the activity matrix, which we use later on in machine learning. And here's already, we could chase some sort of the biological problem of the measurement that activity, especially when it comes to the in vitro versus in vivo. And what are the else competitive mechanisms present in vivo, for example, measurement, and how do you characterize this one? And for this reason, machine learning is also can be used because you can actually switch between different mechanisms, and you can train your model to identify when do you need to use one mechanism versus another, especially when you're developing the time or your particular, if you're in a particular set of the process. And there are obviously well-known certain mechanism, and you can take the correction from that mechanism when you make the assessment of your final activity matrix, especially when it comes to the live, cell line in vivo prediction, etc., compared to in vitro one. So for that reason, once you get that matrix, a correction matrix, you have the tree of a human kinome. So you can formulate the equation and you can perform combinatorial combination with machine learning to predict different activities of the cocktail of drugs. So that shows you basically the activity of the drug across the different concentration and kinesis. So you basically combine two drugs with different concentration, and you see how do that mixture of the drugs impact the different kinesis in the kinome tree. And then by doing these things, you can obviously identify some of the situation so that you can improve the effectivity of the treatment, versus if you just administrate it to one particular drug at certain concentration. And obviously, you could compare your machine learning prediction of the activity versus blind experimental tests. So that, for example, you see the theories dot and experiment was blindly performed for the 2 drugs which we mixed up, and then it was profiled against the kinases. And they see, like, qualitatively, you can, using machine learning, pretty good reconstruct the activity. Obviously, it's not spot on, but there is a lot of assumptions here. But overall, qualitatively, it looks very good. So where's the machine learning comes? So once you administrated certain drugs and you're measuring the healing process, you have usually a connectivity between phenotype and the concentration of and then the drug and the concentration. What you're trying to achieve, you want to achieve some sort of functional relation between phenotype and the kinases, and you want to have the weighting factor net to the kinases to see which one is the most important one. So obviously, you can do the loss regression analysis. You can eliminate the drug as a perturbation mechanism. You can use, of course, you need to use the activity matrix to give it here, and then you can obtain those coefficient beta and beta 0 and beta, which is basically your weight. You show which of the kinases contribute. Of course, here there's a lot of uncertainty, because even if you know what is your competitive mechanism, you still don't know what, for example, the ATP concentration in the cell. And that's a kind of, there are different strategies for a different cell line, and you can basically perturbate that concentration to see what would be the list of the kinases which comes out of the machine learning. And I wanted to show, first of all, of course, how good is your prediction, so which is, not bad. And secondly, if you perturbate, for example, ATP concentration, you could have a slightly different kinases list, which impact your phenotype of interest. In that case, for example, the healing rate of the wounds. And look into those kinases, you can see that, for some of the situation, you can statistically build the kinases, which is most likely, highly likely, will contribute significantly towards that process. And then you can start building up your drug or cocktail of drugs, which will target the particular kinases. So that's kind of ends my presentation. So, and if you have any questions, I will be very happy to answer them. And thank you so much for your time.
Thank you, Sasha. All right, if you want to raise your hand like before, or put your questions in the Q&A or chat. Okay, Maxim.
Yes, Sasha, great presentation. Thank you so much. I have a quick question about polypharmacology that you did with ML. So you showed a slide about the different targets that can be affected by one drugs or some of drugs. So that probably suppose that like drugs can compete on the same targets, taken to the affinity to account to the affinity. Did you consider the pharmacokinetic interactions, for example, acceleration of metabolism, inhibition of metabolism, because that also would affect engagement?
No, we didn't consider that. We've done a very simplistic mixing rules. All those aspects are obviously important. That's why when you compare, let's say, that curve, which shows, our prediction versus experiment. So yes, you see that you kind of like, not too far qualitatively, but it's still, room for the improvements, right? But at least it tells you, like, instead of maybe wasting time and trying to develop the most sophisticated model, you can probably go in that point and then actually test that combination of 2 drugs in a laboratory, right? And see where it works or not, because at this point, that's much faster versus like developing the model itself. Unless if you see that the application of this model later on, for whatever reason, and you need it, right, then you would spend time to do it. At this point, it's more like a fast screening tool to see where you're like in the right direction or wrong.
Right. So this is not about drug-drug interaction prediction, but mostly about the efficacy of drug combination.
Exactly.
Thank you.
Go ahead, Michael.
Okay, thank you, Sasha, for a nice presentation. So my knowledge in the machine learning is very minimal, actually close to 0. But I think I understand the concept in general. I just want to make sure. So suppose we have a kinetic system, right? Described by the equations, PK, PD equations, whatever, right? And they have certain parameters. Yes, it's a good example. Yeah, and this system has parameters, right? And then we have other sources like imaging data, some other covariates, biomarkers that other speakers talk to. So using machine learning technique, we can actually combine this whole flow of information and this sort of optimize, right?
So yeah, so basically it's important to understand what do you mean by combine. So combine means that this is a operator is, for example, what you give it to me.
Yeah,
right?
Constants, for example.
Yeah, so you said that
Rate
this
constants.
is your model.
Yeah.
And you also give it to me YT, right? Which is your image signal, etc. right?
Yeah.
So your system X is a concentration, right?
Yes.
So what I'm building the machine learning, which is mapping from this data towards your concentration. This is a forward data. Where's the combination comes? The combination comes by checking those two conditions, right? Especially this one. So you don't have any control parameters in your system because you don't actually do continuous injection or whatever. You just want to make sure that whatever the data you build, whatever the mapping you constructed from the machine learning point of view, allows you to say whether your system is observable or not. And if it's true, then you can argue that whatever the model you give it to me, a PKPD model, is a consistent, you can carry on doing the simulation and it will be in consistency with any observation data. But most likely you don't have that condition realized. And then you start playing what you can drop on, what you can adjust in your operator A to make sure that, it does fits the rest of the data you observed. So, and that will be basically now a consistency check or sanity check where all the data you have for your study does satisfy the model you built. Otherwise, you could be completely operating in two different worlds, trying to build the machine learning from Y to X without doing the PKPD. And you can do the PKD without actually looking where your Y to X does exist in match, right? So you're trying to put them together and check where this condition is actually true or not.
Yeah, I'm wondering how large this data sets have to be.
Of course, it depends. The C operator depends on the machine learning model you picked up. For example, imagine that I take the random forest model, which Murli mentioned in his presentation, and it has certain, parameters measured like, ratio, some classification where the patient has some of the disease, right? And then basically, imagine that I construct that random forest model, which maps basically my state vector into a certain classification where it has disease, no disease, etc. So the question, that data, that is a huge data, right? I mean, because, we have a multiple big data sampling from the population. It's a huge database, and that operator built in place. Now, imagine that you decided to make some PKPD for that type of the disease simulation and check certain aspect how the drug would propagate and what would be the concentration, giving that information. So effectively, you can combine them together, but you need to make sure that they do exist, they can exist together simultaneously. Why that compatibility condition, let's put it this way, right? So that allows you to say, like, I'm not contradicting anything by bringing those two sets together, right? And it's not trivial exercise, given all those aspects you need to build up, right? You need to, first of all, build the machine learning map data. And you see, like from early presentation, there is a lot of aspects involved. And as well as from your side, on selecting operator A, there is a lot of try and error approach, as you mentioned. And then the question is like, once you're happy, once someone who's processing the data measurement is happy, do you actually can live together?
Yeah, agree. Yeah. So maybe related question to the size of the data. So suppose your system is very noisy, and by definition is very noisy, and that's probably the question to controllability, right? So how can you make sure that that the response that you see, it deviates because of some systematic bias or reasons, or it just uncertainty or error of measurements or any other variability, biological or?
Well, that can be definitely studied via, okay, let me go to this first slide. So obviously, assuming that I wanted to put some noise, model the noise in that additional term via control parameters which obviously depends on X, right? So that effectively will be a stochastic ODE you wanted to solve. So the question, this operator, if you can represent that, you can check where it is controllability, meaning that by the noise, can you stay still, you know, put in the right trajectory, or it's completely will blow up and will deviate it, regardless what is your noise, it will be moving away, right? So that can be done by this analysis, or it could be separately separating the noise from, so you can split the operator A from the median one and then plus noise, and you can directly study, you know, your mean operator, and your contribution for the noise could be just added later on as an additional things, right? You know, multiple ways here. I don't think so I have like the solution in terms of uniform solution. Like obviously there is no free lunch here in terms of there is no unified approach you can pick up and then use it for study. It depends on the application, depend on the what is the noise and variability, where it comes from. Uncertainty analysis quantification will also help you, for example, to identify which of the compartment is sensitive towards the perturbation or not. And then maybe you could argue that you can just based on that criteria can drop and reduce the noisiness in your solution afterward.
Okay, thank you. Yeah.
Okay, any other questions for Sasha? I don't see any in the Q&A. All right, great talks today. Let's go to the Q&A at the end. We may have exhausted all the questions, so as if any come up, we'll get to those. I have a question for the speakers and the panelists. I think what we heard today, again, with any machine learning approach, you know, something I did for years and back at my days at Merck, et cetera, is what I call model quality data, you know, having enough of it. So I think today we kind of saw that a lot of this modeling is dependent on data and model quality data. We're seeing companies that are starting to collate and curate data for a living, and I think this is going to continue. But I just kind of would like to hear everybody's thoughts on maybe what you think we should be doing differently from a data perspective and what I call model quality data. So whoever wants to start, just go ahead.
I can start.
Okay.
I mean, obviously, curating the data is an important step. However, it has its own caveats in terms of who is doing, what is the reason, and what is the purpose, right? You know, and if it's transparent and clear, and then whatever the attribute or metadata assigned to the data you curated, that you can actually revert it back if you're not happy with some of the assumptions behind that process, that can restore the original data, I'm perfectly fine. But I would rather maybe keep it, you know, as it is, whatever the data is available. And maybe it's more related to fairness of the data. We already discussed with you, John, on a couple of the webinars. If it's transparent and clear, and you know it addresses the methodology how the data was obtained, I think the curation process is important, but it can be done separately by each of the scientists or application, whatever it is necessary. The tendency, which I've seen sometimes, the curation of the data lost their very important details, which is very difficult to track it back. And I think that somehow needs to be acknowledged. And of course, you know, you don't want to release only whatever you wanted to release for the sake of the demonstrating that, you know, only successful stories. You wanted to release all the data and all the results, make sure that you can have a proper assessment of the entire the simulation pipeline of predictions, right? So those would be my thoughts on that question.
Okay.
And if I can jump in, I sort of second what Sasha just said. I think the key issue is that you really have to be very aware of observer bias, right? And so I think that we all collect data for our own purposes with our own biases. And the whole point of investigating data is twofold: to find things that you didn't know before and you did not expect, which is a contradiction, because you really also want that same data set to sort of validate something that's already known. And these two things are in direct conflict with each other. The surprise element and the comes from finding something new that you can test again and make discoveries around, and the validation element, which is from finding something that you already know, that builds confidence on your new discovery. So the two elements have to be in the same place, and I don't know how you actually address it effectively.
So, Dr. Morale, I'm not picking on you, right? But my question is, academia, could some of this be taught in school? And I'm not saying even college or graduate school, but even earlier, right? The whole scientific method, the need for the scientists to be more aware of data as an asset, you know, that ground roots type of approach. Because after my 30 years of doing this, I found that it's a cultural problem in R&D, right? That we don't have this mindset, like, you know, we have it with money and all, you know, this data is just converted currency, right? But we don't have it with our data. And I feel this is the root cause of the problem.
I am sort of going through my own, pardon me, I'm an academic, so I tend to sometimes become philosophical, my own experience with data. And if you look at the traditional trajectory at which, from which many of us grow into data, it is typically starting out with arithmetic and mathematics, and maybe progressing into statistics. But all of us actually grow the most when we experience problems and we experience data on our own, because then all the statistics and the mathematics and everything that you invested in suddenly begins to make sense, because it's personal, right? Yeah. And I don't know how we can go. And I think that data intuition is another thing that many people, many of us who have experience have developed. We don't need to dive into every spreadsheet and every column of data to figure out whether the integrity has been compromised in some way. That is because we know our domain so well, right? You know, maybe there's that EC50 makes no sense, or maybe whatever it is that we use as a heuristic. So those are the two things. I think there's the intuition and there's the experience that teaches you about the method. And I don't know how to approach it, but I think that it's going to become important and very fundamental. Maybe college is the place to start.
Okay. Anybody want to comment on that?
Maybe I can end on that one. We don't want to be trapped ourselves in a situation when everything will be data driven, right? So I could imagine the situation when even multiplication of two matrices at some point will be just statistical. What if I'll multiply two matrices? Okay, that looks like this answer with probability of 95%. We don't want to be in this situation, because those are things that are deterministic. We need to teach, we need to know that the people understand the things coming from. It's important because any data analysis, I mean, Murali mentioned one of the very important, the no free lunch, but there is another one, garbage in, garbage out. You need to make sure that, you know, the domain knowledge is still there. You understand what they're doing with the data. And without that, you know, you could do all sorts of crazy things, right? So even it is data driven. So therefore, like, the data has to come in the right time when the person is already know and has a certain fundamental knowledge before actually start operating and relying on those tools, because they could be very dangerous equally. And used in a very different way.
Yeah. Okay. If Michael or Maxim, you have any comments, we'll kind of move on to another question.
You know, I have a quick comments. I will join Sasha talking about the data curation, accuracy of this data, because, you know, in my experience, sometimes people, a research team, they curate data for their own purpose and then just release it to the media and to the audience. And I had an experience when we just uploaded like a huge data into the database and then found out that it is not accurate. And so we needed to refine the whole database, trying to figure out what is wrong. So I would love to, to find some source of like accurate data for us, for example, for PK and PD parameters. But on the other hand, it always needs some verification. And it also our own manual work. So I don't know, maybe we can automate it. Maybe just to find some outliers that would show you that this is probably the corrupt source, or this is just accurate data. That would be probably good. Maybe it will be another implementation for ML algorithms to just check all the data, to find outliers, to find the data that doesn't correspond to some patterns, so we can identify them. That's for me, for example, because I'm responsible in our team for the scientific accuracy of the database, to find out what data is correct and what data is not, I would say, not so correct.
It actually raises a very interesting point, Maxim, after your comment. So would it be possible at some point to have all databases are open so that the crowdsourcing activity can actually clean this up? Because, obviously the manpower and the crowdsource is much huge compared to any other activity. So they can figure out quite easily, someone find the spare time to check and say like, oh, this is wrong or something like that. But the question like, where we can achieve that moment in time, because the data in most of the companies is treasure, that's what caused their asset, and they rely on some of the product based on the data, and it's really hard to actually combine. And that's the reason the federated machine learning come into the game, when you cannot actually get access to the full data across different providers, but you're trying to train your model, but you don't know how the data was created in the first place, who was behind the scenes, for that particular database, and that could potentially lead to, a wrong conclusion, right? So that we're trying to avoid.
Yep. Well, I mean, it's like other areas, and Michael, you probably want to comment here too. You've got standards, you've got what Murali brought up before around, metadata. All these things kind of come into play. There is a high level of complexity, I think, in the environments that you guys are working in, compounded by the algorithmic approach, training sets, etc., versioning models, all that comes into play here. There is a level of complexity here. But I think based on my experience I walk into 80% data wrangling environments. Like I have people that are so uptight sometimes because of their data environments. They don't know if they can use data. They don't know, it's not fair. They don't know if they can access it. They don't even know about it. And then many times you have competing or opposing camps in these areas in large pharma. And it's like a really big, tight knot. You have to figure out how to undo it and kind of level it and start over. And that's some of the experiences that I've had.
So if I can jump in, John, my personal feeling is that creation of large data sets is best done when it's planned and centralized. Sometimes, a lot of the times, the temptation is to agglomerate data, aggregate data. Oh, so investigator X has, 200 MS patients, and investigator Y has 300 MS patients. Let me put it all together. Obviously, that gives you a jump start. It gives you a sense for the diversity and the heterogeneity of a few measures, but it does not make for a good process that doesn't require data wrangling. Let me put it this way. On the other hand, I have gone into, as you notice, I tend to focus a lot on government sources of data, and the quality of some of those resources is just remarkable. I can tell you that in the private sector or in academia, any effort to collect data is not fundable. So if I was a clinician and I'm just making this up, if I had the entire United States as my patient population, I still would not be able to find folks who do the data collection, the data entry, the quality control. There would be no way I could fund that.
Mhm.
But on the other hand, the government has developed processes that are quite getting better with time.
Yep.
And that you can sort of see. I go into the NHIDs all the time. I've gone into some really big data sets. And you go, on the other hand, to some scientific papers, and people put out quote unquote data into public repositories. The quality goes downhill really fast.
Yeah.
They get large, very heterogeneous, but you really don't know. The QC is just so poor in repositories. It's crowdsourced, I mean. And so that's my general sense. So I go into these, I avoid repositories if I can. Let me put it this way.
What are your thoughts, Michael? I know you've been off mute for a little while, so you're probably trying to get a word in.
Yeah, so well, my problems are usually that I'm currently working on, they're based on small data.
Mhm.
So my problem is actually getting the data itself. So is that biology is not understood, mechanisms are unknown, we're just guessing. So based on some preclinical experiments and some very, very limited clinical data. So that's the
Right.
problem.
There is a phrase out there: big data gets all the press, little data does all the work.
Yeah, that's yeah.
It depends who you are and what
yeah,
you're doing, but
yeah. Yeah. So typically for constructing our models, I mean, the model that I illustrated, so it comes from like 30 animals, 30 plus animals, 40 animals, and it's just maybe dozens of time points, essentially, right? So we're trying to build relationships through this data. And there are other sources like imaging data, but those are unreliable and qualitative rather than quantitative. So we don't know how to interpret this type of data. I mean, we know, but we take it with a grain of yeah. So, and of course there are like in vitro cell type assays data, and of course there are bioinformatics data on relationship between genes, associated with certain diseases. So yeah.
Do you guys see more technologies in the near future, like organ on a chip, or different approaches where you think you might be able to enhance your work, or do you think it's going to be a slower move forward? Or is it a dumb question?
I mean, in my experience, again, what I see is a sort of it's by stages. So it's like first bioinformatics groups identify targets and potential molecules, then preclinical experiments confirm or basically validate the hypothesis, and then preclinical data being generated, like PK and pharmacology data, and then we build some kind of PK/PD models and test it in human. That's the process, right? So given all other limitations like safety and toxicology and agency regulations. Yeah.
I hate to be the bearer of bad news, and you all know this, but small molecules make some progress. Large molecules are hard in this space, right? And I think this is where we need new discoveries. We need new ways of approaching things. Any comments on that? Alex, you agree or disagree?
No, I agree. That's the reason we actually kicked off the discussion about the machine learning and data-centric QSP, because I think that's one of the opportunity to think a little bit out of the box and then trying to apply various sorts of information. And then obviously discoveries comes in, more smart drugs will be developed, because they will be like really specialized, take into account certain aspects, rather just be pretty much every step across the population, and then have a very coarse type of treatment plan. So I think it's not only combination of the molecular structure, right, small versus large, but also how do you administrate it, what do you do, like dosage, etc. That all comes to the game, and be like more tailoring towards your concrete personality in terms of like what is your biological system looks like and what do you need, rather just, you know, you and me taking the same in a sense, right?
Yeah.
And see where it works, where it doesn't work. So I think that's where the advantage is going to be made in the future. So again, you know, combination of various of drugs, etc. That's something, you know, actively exploiting. That also potentially could lead to. And actually, if you look at the history of some of the very impressive drugs, you know, they usually discard, you know, very unaccidentally, right? So completely not, you know, working in that direction, right?
Yeah.
Like develop for something else and suddenly it started working elsewhere, right? More effective. So,
all right. Any questions from the panel? Do you guys want to ask a question that maybe I didn't ask or you were thinking about that you would like to get everyone's opinion on?
maybe I can just say and thank everyone for accepting the invitation to come to the webinar.
Yeah.
You know, really great pleasure to listen to the talks and having a discussion with you and hear some of your opinions. Obviously, we'll keep pushing that initiative to organize maybe like on a yearly basis some of the events like that, and hopefully it will become like a small local chapter, and we'll see the progress from all the participants, right?
Yeah. Yeah, don't give up the fight. It's a noble effort that everybody's doing, and it's pretty awesome. And it was great to see. I did some. A colleague of mine published a paper years ago at GSK on Bayesian learning, replacing animals models with Bayesian learning, and it's great to see how powerful the Bayesian models still are. And I think what people that aren't in this space don't realize sometimes is you almost run out of options of what do we do next? We're not sure where to go. And sometimes the models help you go into a new direction, or you say, well, I went in that direction, now I'm going in the other. And so it was really interesting to see the talks today, and I thought everyone did an awesome job.
Thank you.
Yep.
Yeah, thank you for great presentations, and I learned a lot personally. And by the way, I see that this webinar was recorded, right? So
oh yeah, it's going to go on demand. So
okay,
people will register for it and watch it. Yeah.
Okay, perfect. Okay. So then we can always go back and take references, and
yep.
It was very, very useful. Yeah.
Yeah, I think so. Yeah. Awesome. Okay. Well, thanks everybody. Enjoy the rest of your week, Friday, and have a great weekend. And thanks so much. Really appreciate it.
Thank you all.
Stay healthy.
Thank you.
Right. Thank you, Sasha. Thank you, Max.
Bye.
Thanks, everyone.
Thank you, Mariah. Thank you, John. Bye-bye.
Bye-bye.